Good morning, everyone. Today is the second day of our conference. So, honorable dignitaries, respected faculty members, dear colleagues, professors, lecturers, researchers, and academicians. I am Siddhartha. It is our honor and pride to warmly welcome you all the 7th annual meet of International Association of Geologists and 4th International Conference on Research Trend in Life Science and Biotechnology. And this is the second day are organized by International Association of Geologists and CD Earth Group of Education. Our collaboration with Department of Biotechnology Maharishi Markandeshwar deemed to be University Mulana Ambala and Sarda Institute of Science, Technology, Education and Research. The theme for the year 2023 is biological, physiological, chemical and radiological agents perspective to human health and diseases. I honorable welcome our all participants and today's guest speakers, Dr. Tina B. Saini, Dr. Tina Bakuni, Dr. Nanus Safakatullah, Dr. Usman Umar Jango, Dr. Navjeet Kaur, Dr. Aman Giri, and Dr. Seema Makhija. Now, we are welcome the participants for oral and poster presentation. The guest speaker lectures will start approx 9:30 to 9:40 a.m. Before we will proceed the oral and poster presentations. The first name of the participant is Komal, Shreya, Yasika, Kumari Resu, Amita, Manjulan Kumar, Surichi Kumari, Supriya Upadhyay, Avilas Sahu, and Aishwarya. Uh, so the first participant is Komal. First participant is Komal Sahu. Next one is Shreya. Shreya Chaudhary.
Good morning, everyone.
Very good morning, ma'am.
Okay.
Ma'am, please present your paper.
Okay, sir. Just give me a minute.
Okay.
So, is my screen visible?
Yes, very much visible. Please zoom it, the portion,
Okay.
or rotate the mobile.
Okay, sir.
Yeah, that's good.
Okay. So, good morning, everyone. My name is Shreya Chaudhary. I'm from BBAU Lucknow. So, today my poster presentation topic is Talk on Immunology and its Multifaceted Applications. So, firstly, we will start with what is immunology? It is the study of immune system as our immune system protect us from infection through various line of defenses, and it maintains our overall health. So immune system is the major factor for protecting us against the viruses and all type of infections. Now if we talk about cellular responses and defense in animals and plants, so firstly we will understand that what is cellular defense. So cellular defense is nothing but basically it is the another name for innate immune responses that protect us from any type of external infections. Now, it could be of many different types in plants and animal, but generally we focus and talk about phagocytosis and antibody production. So, if it in animals, if we talk about phagocytosis, it generally means that it is a central mechanism in host defense against invading pathogens, as it involves recognition, uptake, and destruction of all the microbe. Phagocytes are nothing. Phagocytes are basically nothing, but they are the type of WBCs which protect us from the external infections by invading the pathogens. and the other one is antibody production. Like if we talk about first line defense, it is phagocytosis, and after the phagocytosis, it is the antibody production. In antibody production response, B cells are activated which secrete antibodies, which are generally proteins. We call them immunoglobulins, and human we have 5 types of immunoglobulins. In G, A, M, D, E, there's a 5 types. Also, these type of cellular responses we can see in graft reactions. When a graft reaction occur, when a recipient's immune system attack the donated graft, in this case we also see the cellular responses by our B cells and the T cells if they don't accept the grafts. Now another topic is immune response to parasites. Our immune responses are basically divided in 2 types. One is specific and the other one is non-specific type. Now, firstly, we will talk about non-specific defense. Non-specific defense mainly contains the physical barriers, inflammatory responses, phagocytes, but whereas the specific cellular responses contains defense by WBCs, clonal selection of T lymphocytes, T lymphocyte targeting immune responses, and destroying infected cell by inducing apoptosis. Now, when we talk about immune responses to parasites, there was a very wide term we use, which is herd immunity. Herd immunity is indirect protection from an infectious diseases that happen when a population developed immunity by previous infection. And when we talk about the previous infections, we also talk about host immunity. Host immunity contains the B lymphocytes, which makes antibodies, as antibody helps body to fight microbes or toxin they produce. They do this by recognizing substances which we call antigen on the surface of microbes. Antibody mark these substances, mark these antigens for destruction, and then it goes for the cell mediated immunity by T lymphocytes. Cell mediated immunity works against infection inside cells. T lymphocyte can recognize and kill infected cell or program the cell to kill microbes inside themselves by producing cytokines. Now, lymphocyte activation is required when we talk about host immunity. Lymphocyte activation occurs when lymphocyte, either B cells or T lymphocyte, triggered through antigen specific receptor on their cell surfaces. They help lymphocyte activation generally helps cytotoxic T cell to kill any kind of infected target cells. Now, the another aspect of immunology is antibody techniques. As we know, antibodies are widely used in detection and identification of specific proteins. So we use different type of antibody techniques for different type of functions. Nowadays, we use antibody in our technical methods, as immunoassay techniques use antibodies linked to reporter enzyme to cause a color change in presence of a specific antigen, fluorescent labeling of antibodies in bloating, also use of monoclonal antibodies in the diagnosis and detection of diseases, use of ELISA technique to identify the presence of specific antigens to produce stocks of a particular antibody, and hybridomas also produced by fusion of B lymphocyte with a myeloma cell using polyethylene glycol, which is PEG, and use of monoclonal antibodies in the diagnosis and detection of diseases uses the ELISA technique to identify the presence of specific antigens. Now, regarding the production of monoclonal antibody, further we use the another mechanism, which is the clonal selection model. This model is given by McFarlane Burnett and the Peter Medawar. They also got the Nobel Prize in Physiology or Medicine in 1960. They, in this clonal selection model, they generally define or term the words like self-tolerance, immunological memory, and vaccine designation for the protection from the any kind of antigens. Now, if we talk about production of monoclonal antibodies, we use monoclonal antibodies for different type of therapies: autoimmunity, inflammation, infection, transplant rejection. These are the different type of antibodies. Now, if we talk about the production, like how we produce monoclonal antibodies, we mechanism by which monoclonal antibodies are produced is called as hybridoma technology. In this, there are different type of steps we use. Firstly, we will use animal immunization, in which we in which we okay sorry animal immunization is a process in which we immunize a particular animal, or basically we use mice with the appropriate antigen or a adjuvant. It enable stimulation of B lymphocytes. Then we it mixed it with HDPRT, basically myeloma cells, and exposed it to the PEG system, which is polyethylene glycol. Now the system, now our system contains the free myeloma and free lymphocytes, which are the few cells. Now the cell will cultured in a hat medium, and in a hat medium, it is a specificity that only hybridoma cells grow. So now we select the hybridoma and make antibody specific cells for the antigen that we put in the mouse. So this procedure is called clone selection hybridoma technology. As we know, monoclonal antibodies are very therapeutic advantages, as they block biological function by neutralizing soluble factors for inhibiting cell-cell interaction, killing target cell through receptor blockage, activating cytotoxic immune cell by selecting targeting of a cytotoxic compound. As one of the major example of monoclonal antibodies for therapies is given on the right side. Like approximately 20 to 30% of breast cancers overexpress the human epidermal growth factor receptor 2 on their cell surface. So for that, a trastuzumab is a humanized monoclonal antibody that bind to this particular growth factor and stop dividing of cell. It also suppresses angiogenesis. Angiogenesis is a blood vessel development. So this is how our monoclonal antibodies protect us from the breast cancer and minimize its growth. Okay, thank you. This is my presentation.
Can you present your title?
Yeah, wait.
First thing, when you present a poster, the title is not good because the talk on immunology and its the talk word is not used in the poster or in the oral presentation.
Okay, sir.
Is virus cause a cancer?
Can you please repeat?
Is viruses cause a cancer?
Viruses cause a cancer?
Yeah. Is this statement is true?
Uh, yes, sir. Like HPV for cervical cancers.
Okay. Okay. Thank you.
Okay, thank you, sir.
Next one is Yasika.
Hello, good morning, sir.
Very good morning.
Good morning to
Please
all the.
present.
Oh, yes, sir.
Next one is Komal Sahu.
Sir, just a second. Sir, is my poster visible?
Yes, yes, visible, very much visible.
Okay. Good morning to all the esteemed guests and respected teachers. Today, myself Yashika from BBAU University Lucknow, I am here to present on the topic antigens and products derived from helminths and their impact on type 1 diabetes. For this, we will firstly discuss what is type 1 diabetes. It is a chronic metabolic disease characterized by elevated blood glucose levels as a result of insufficient insulin production by beta isolate cells of the pancreas or impaired insulin sensitivity of insulin target organs. According to the International Diabetes Federation, around 387 million people were living with diabetes in 2014. Diabetes incidence is increasing both in developed and
Please
developing
zoom,
countries.
please
Yes,
zooming
sir.
the PPT. Rotate the mobile and yeah,
Yes, sir. Is it okay, sir?
yeah, yeah, it's okay.
Okay. So, diabetes, as we discussed, is a chronic metabolic disease described by eminent blood vessel level as a result of insufficient insulin production by beta isolate cells of the pancreas of insulin target organs. According to the International Diabetes Federation, around 387 million people were living with diabetes in 2014. Diabetes incidence is increasing both in developed and developing countries worldwide, and it is predicted that in 2035 up to 592 million people will suffer from diabetes. It is an inflammatory immune responses that play a crucial role in the progression of beta island cell destruction in both type 1 and type 2 diabetes and insulin resistance in type 2 diabetes. Conversely, helminth infection are well known to induce type 2 and anti-inflammatory immune responses that are able to counter-regulate inflammatory immune responses that will lead to diabetes onset. For this, we need to know what is type 2 immune response. The T helper type 2 immune response is characterized by the production of interleukin 4, 5, and 13 and is a critical immune response against helminths invading cutaneous or mucosal sites. Now we will discuss about the inverse correlation between helminth infections and type 1 diabetes incidence. So type 1 diabetes is an autoimmune disease which, as we discussed earlier, the subsequent loss of insulin production leads to increased blood glucose levels that led before the discovery of insulin by Banting and Best in 1922. Especially in developed countries, the incidence of type 1 diabetes is dramatically increasing. This is because genetic factors alone cannot explain the strong increase in diabetes incidence observed over the last decades. Environmental factors have to be taken into the consideration. One major change in the surrounding environment of people living in developed countries is the improved hygiene. And because of improved hygiene, and because of improved hygiene, the bacterial, viral, and parasitic infections have reduced. And with regard to parasitic helminth infections, it can be assumed that the immune system of humans evolved in the presence of parasitic helminths, as human remains from Neolithic and pre-Columbic Americans showed helminth infections. Still, during the first decades of 20th century, helminth infections were very common. And for this study only, scientists have introduced an important tool in the research of TID, T1D, that is type 1 diabetes, and it is non-obese diabetic NOD mouse strain, which was generated in 1980 by Makino and colleagues in Japan. Particularly, female NOD mice are prone to develop T1D with around 80% probability. Before clinical signs of T1D become independent in NOD mice with glucosuria, hyperglycemic, and substantial weight loss. Mononuclear cell infiltration to the parasitic to the pancreatic islands occur. This infiltration can already be seen by the age of 6 weeks. Several cell types are known to be crucial for the initiation of the autoimmune process. Macrophages and dendritic cells, as well as B cells, have been found in the pancreas of pre-diabetic NOD mice and have been associated with autoantigenic presentation and expansion of autoreactive CD4 plus and CD8 plus T cells and destruction of the insulin producing beta island cells, initially leading to insulitis and subsequently diabetes onset. Therefore, the scientists concluded that diabetes induction in the NOD mouse model is similar to the human pathogenesis and suited to examine the influence of infections. That's why it is considered an important tool in the research of T1D, that is non-obese diabetic NOD mouse strain. For this, we will discuss the helminths and their effect on type 1 diabetes. There are several helminths and they have different types of effects on diabetes, and all of them work on different possible mechanisms. For example, first parasite that we are going to discuss is H. polygyrus. Its effect on diabetes is prevention in NOD mice with 4 to 6 weeks of age. Delay in NOD mice 7 to 12 weeks of age, reduced hyperglycemia in multiple low dose streptozotocin model, and its possible mechanism is shift to type 2 immune response independent of CD4 plus CD25 plus T cells and interleukin 10 signaling independent of STAT6. For this, we need to know how STAT6 works. STAT6 activation initiates a transcription program comprising both immunological effector responses and metabolic adaptations necessary to sustain them. So, IL-10 signaling required in the absence of type 2 immune responses. That is how H. polygyrus works. Next, L. sigmodontis.
Please conclude.
Okay, sir. For this, we need to know how helminth-mediated protective mechanism basically works. How we can protect the onset of diabetes with the help of helminth. Possible helminth-mediated protective mechanism that prevent the onset of type 1 diabetes, infections with living helminths, administration of helminth egg as well as helminth antigens can induce type 2 immune response. T helper type 2 cells will produce interleukin 4, 5 and 13 and further promote the type 2 shift and counter-regulate activity of diabetogenic autoreactive T helper type 1 cells. IL-4R receptor signaling further induces alternatively activated macrophage, that is generally known as AAM differentiation. Helminths and their products additionally derive regulatory anti-inflammatory immune responses by inducing AAM regulatory T cells and tolerogenic dendritic cells. IL-10 and transforming growth factor beta, that is TGF beta, derived from AAMs and regulatory T cells can inhibit the activity of autoreactive T cells and suppress CAM, that is classically activated macrophage induction and natural killer cell activation. Taken together, induced regulatory and type 2 immune response that are generated due to the invasion of helminth suppresses the diabetogenic autoreactive T cells, natural killer cells, that is NK cells, and CAM, leading to a reduced loss of beta islet cells that will result in decrease of diabetes. Thank you, sir. That was my poster.
I have one question.
Yes, sir.
From figure 1, which type of response, excessive inflammatory response on the host cell or reduction the macrophage activation? Which
Sir,
type of
can you
response?
please repeat? I didn't understand.
Which type of response in figure 1, which type of response promote a reduction in the macrophage activation?
Okay. Just a second, sir. Macrophagic response.
Which type of response? This is which type of immune response?
Sir, innate immunity. Type 2 innate immunity.
Okay, but response type is okay. Leave. What is the difference between type 1 and type 2 diabetic?
Type 1 and type 2 diabetes.
Yes.
Sir, type 1 is autoimmune disorder in which our body kills its own cell. That is why it is it is considered as
You have written in the fourth line in your
yes, sir.
abstract or introduction.
Yes, sir.
Abstract in fourth line you can written.
Yes, sir. Helminth infection. Yes, sir. Induce fourth, sir, in the first paragraph.
Yeah, first paragraph, fourth line. The pancreas does not make insulin in type 1, and in type 2 pancreas less insulin. Okay, leave.
Yes, sir. That is what I'm explaining. Diabetes type 1, the pancreas does not make insulin because the body attacks its own cells. And in type 2, the pancreas makes less insulin than used.
Okay, good.
Yes, sir.
Next one is Kumari
Okay.
Resu.
Thank you, sir.
Thank you. Next, Kumari Resu.
Yes, sir.
Yeah, please.
Yes, sir. अम्मा कैसे होगा? अम्मा नहीं रमा नहीं हो रहा है आज. So is this visible?
Yes, visible.
Presentation. Okay. Thank you. 1 sec. Good morning, all of you. My name is Rishu, and I'm PhD scholar, pursuing PhD under the supervision of Dr. Nishma Jaiswal, Babasaheb Bhimrao Medical University. Today, I'm here to present my presentation on the topic establishment of Decnatoides species from marine host fishes C. modetta and L. malabaricus to freshwater host fish Rita by means of bioinvasion. In this presentation, I would like to go with the abstract part. Abstract is this, and after that, introduction is there. In this, India ranks third in the production of fish in world and ranks second in the inland fish production. And catfishes are distributed all over the world, often a component of human diet. Catfish oil are considered to contain PUFA, where the percentage of PUFA is low in the catfishes as compared to the oil extracted from the marine fishes. Basically, what I'm going to talk is about the bioinvasion. Bioinvasion or biological invasion is the establishment of non-native species outside their natural range, which represents a major threat to a diversity, integrity, and functioning of ecosystem. Next is the material method. The material method which I followed is that the host fishes, my host fish was Rita Rita, which was collected from the Ghala Bridge of Gomti Riverine ecosystem. And after that, the fishes were collected and transported to the laboratory and checked for the parasitological presence. For after investigation, we got the nematode parasite, and nematode parasite were further processed for morphological analysis and molecular analysis. For morphological analysis, we pursued light microscopy and scanning electron microscopy. Light microscopy, for light microscopy, we followed these protocols, and scanning electron microscopy, we followed these protocol. Molecular analysis, for molecular analysis, DNA extraction was done using Kaizen, DNA, blood and tissue kit. PCR amplification was performed. After that, the PCR products were sent for the sequencing at the Future Biotech Bangalore. And one sec. Result. My result is that light microscopy. For the light microscopy, I got these pictures. First picture represents the interior end of the female nematode. Second picture represents the vulvar region of the female nematode, and posterior end of the female nematode, and anterior end of the male nematode. Fifth is the posterior end of the male nematode. In the fifth picture, there's a cephalic alley called a cephalic alley present in the posterior end of the male nematode. And in the same pictures, I got these pictures. Figure 6 represent the anterior end of the cephalic, anterior end of the male nematode. Here the cephalic teeth are present, and in the figure 7, anterior end of the male nematode is shown. Here the cephalic teeth are shown. Here the cephalic teeth are shown, as well as the sensory organs are also present. Here it is a amphids. Amphids are chemoreceptor, which are present at the anterior end of the male nematodes. The figure 8 represents the posterior end of male nematodes, and it is a lateral view of the posterior end of male nematode. Figure 9 represents the posterior end of the male nematode, and it shows the phasmid, presence of phasmid, which act as a sensory organ in the posterior end of the male nematode. Figure 10 represents the female nematode, posterior end of the female nematode, lateral view, and in this structure, sensory organ, that is DRID, is present, and which act as a sensory organ in the female nematodes. And molecular observation, for molecular observation, I got these sequences, which represents the Decnotoides species of from Rita Rita of Gomti riverine ecosystem. My finding is that after that the phylogenetic analysis was done. I constructed the neighbor joining tree using Mega 11, and in this in the phylogenetic analysis, it was found that the nematode of Rita Rita, which is found to be Decnotoides species of Gomti riverine ecosystem of Lucknow, show 100% similarity with the Rita Rita of Ganga River, Allahabad, but it shows 99% similarity with the Decnotoides species, which is also present in the marine environment of center-west coast of India, of host fishes Corynmus modatta and Lurjanus malabaricus. It shows 99% similarity with the Decnotoides species of Gomti River, as well as Decnotoides species of Ganga River. So the next thing what comes in our mind is what is the reason behind that similarity and dissimilarity. The 1% dissimilarity is caused due to the adaptation of marine parasites to the freshwater ecosystem, that lead to the 1% dissimilarity, but the 99% similarity was found, which is due to the bioinvasion, because the marine species Decnotoides species, which was retrieved from the marine fishes Corynmus modatta and Lurjanus malabaricus, was transferred to the freshwater ecosystem by a process of bioinvasion. The bioinvasion is due to the ballast water, which is transported from marine to freshwater uh, during the process of shipping. So in 1985, under the Act of IWAI Act, Inland Waterways Authority of India was constituted, whose function was to develop and regulate inland waterways for shipping. So in October 1986, National Waterway 1 was started between Prayagraj and Haldia. So whose length was 1,620 kilometers. So for that sake, during the process of Shipping, the ballast water which is filled at the port of Haldia in the ships is transported to the freshwater ecosystem of Ganga or Ganga river ecosystem. So it is shed off in the freshwater ecosystem. So the mixing of the marine species with the freshwater ecosystem, freshwater species took place, and with that process, the bioinvasion occurred in the Decnotoides species. National Waterway 1 connects the Prayagraj and Haldia. Cargo transportation and commercial shipping vessels movement was also performed, which led to unintentional dispersal of invasive species between marine and freshwater ecosystem due to unmanaged and untreated ballast water released in foreign ports, which could potentially introduce a range of marine species. And ballast water often spreads 7,000 living species to new habitat across the globe. It was found by the Ilsic et al. 2013. Bioinvasion of parasite causing disease in fish fauna is one of the foremost danger to the aquaculture industry. It is considered that improper treatment of ballast water or improper tech or not full development, not proper development of the techniques for the treatment of ballast water during the during the shipping voyage is one of the most important cause because behind the bioinvasion of the helminth parasite of different species of different fish species. In the present study, the phylogenetic analysis showed that parasite species of Decnetoides species from Rita show a significant similarity index with those of marine ecosystem of Arabian Sea as well as freshwater ecosystem of Gomti River, Lucknow and Ganga River, Prayagraj. Hence, it can be concluded that bioinvasion is the driving force behind this type of migration of same species in two different habitats. The present finding will help us significantly in the studies related with the host-parasite relationship and their host and their role in the environment, disease, and physiology. Thank you. These are the references which I followed. Thank you.
Thank you, ma'am. If anyone asks two questions, then please unmute yourself or raise your hand.
Yes.
No. Next one is Amita.
Yes, sir.
Yeah, please present.
Sir, my slide is visible.
Yes, very much visible.
Good morning, everyone. My name is Amita Singh from Baba Saheb Bhimrao Ambedkar University, Lucknow. Today, I am going to talk about my topic to explore the dietary habits of lesser mouse-tailed bat Rhinopoma hardwicki in Barabanki, Uttar Pradesh. Introduction. What is diet? Diet is mainly kind of food which is an animal habitually eat and which is gives us nutrition, like we eat fruit, vegetable, legumes, etc. On the basis of diet, bats are mainly two types: frugivorous bats and insectivorous bats. And Rhinopoma hardwicki is also is an insectivorous bats. The little bat Rhinopoma hardwicki has a free tail, a small nose, and leaf-shaped snout. Its color is gray to dark brown. Large colonies of Rhinopoma hardwicki can have anywhere from a few hundred to several millions of individuals. Insectivorous bats provide important ecological services such as pest control. पेस्ट कंट्रोल जैसे सर जो इंसेक्ट्स हमारे क्रॉप्स को डिस्ट्रॉय करते हैं जैसे टोमेटो कुकुम्बर एक्सेट्रा, उन इंसेक्ट्स को ये बैट्स खाते हैं. The aim of this study is to provide insight into the feeding habits of Rhinopoma hardwicki in Barabanki. Material and methods. Study species. Lesser mouse-tailed bat Rhinopoma hardwicki has most recently been assessed for the IUCN Red List of Threatened Species in 2016, and now Rhinopoma hardwicki is listed as least concern. Classification. Uh, Domain Eukaryota, Kingdom Animalia, Phylum Chordata, Class Mammalia, Order Chiroptera, Family Rhinopomatidae, Genus Rhinopoma, Species Hardwicki. Study area. The study was conducted in Daryabad district, Barabanki, Uttar Pradesh. This is the roosting site and this is bat colony. Sample collection. Bats were captured using mist net. Their sex and age were noted, and their measurement were also taken from the bat for the species identification. And 30 to 50 fresh feces were collected beneath the colony roosting area of the species by placing captured bat into holding bags. The feces were stored separately in airtight container. Diet analysis. Guano was taken to the laboratory, dried and stored at room temperature. Before analysis, the droppings were soaked for a few minutes in a mixture of water and ethanol. All possible identifiable items were removed, mounted on a slide, and examined under a microscope. This is the guano. And sir, this is the procedure which I can use. And this is the stereo microscope. Result. Total 192 pellets were analyzed during study period September to November 2023. The findings were shows 3 order, 5 families. 3 order Coleoptera, Hemiptera, Orthoptera, and 5 families are Carabidae, Heteroceridae, Scarabidae, Pentatomidae, Ligeidae. This is the all pictures which I have found during my observation. Thank you. Hello.
Thank you so much. Now next participant is Manjulan Kumar.
Yes, sir.
Yeah, please proceed.
Sir, just wait a minute. Entire screen. Sir, wait, sir. Actually, I'm making a PPT upload. देखिए सर विजुअल
विजुअल विजुअल हाँ है विजुअल
ओके सर my PPT is visible.
Yes, visible.
Okay sir, my topic is assessing the contemporary landscape of lymphatic paralysis in India. Actually this topic is given. So I'm presenting here. My name is Manjulan Kumar, PhD scholar, Baba Saheb Bhimrao Medical University, Lucknow. This is my abstract. First of all, we are going to introduction. The paralysis has been a major public health problem in India next to the malaria. The disease was recorded in India early 60th century BC by famous Indian physician Susruta. The 7th century AD, Madhavarka describes signs and symptoms of this disease. It writes Madhava Nidhana, which hold good even today. In 1709, calls called elephantoid legs in Cochin Malabar legs. A discovery of microvilli in peripheral blood was first by Lewis in 1872 in Calcutta, now in Kolkata. Lymphatic filariasis, often referred to the elephantiasis, is debilitating and neglected tropical disease. Uh, this disease can to lead severe disfigurement, disability, and socio-economic consequences for those affected. First of all, we got also know the causative agents of the vectors in India. In mainland India, major illness accounting the 99.4% of issues is caused by bacteria of Rhinophyta. The common vector of disease, this parasite is Culex cunifasciatus. Second one is Burgia malai, which is not much more percentage in India. So the mosquito species Aedes nivis has been implicated as an infectious vector. Transmission of the lymphatic filariasis. Figures show the adult filariasis worm microvilli in hepatic lymphatic system of men. In India, 99.4% of the cases are caused by species Bacteria Rhinophyta, whereas Burgia malai is responsible for the 0.6% of this problem. The in adult stage, filariasis form live in the vessels of lymphatic system. Lymphatic system is the network of lymph nodes and maintain fluid balance between the tissue and the blood, which is essential for the element of the many things. This is a life cycle. The definitive host is the man. Man is the definitive host of the Bacteria Rhinophyta. The adult parasite usually found in the lymphatic system of the man. They give birth as many as 50,000 of the microvilli per day. And parasite life cycle also in the mosquito begin with the microvilli picked by the vector mosquito during the feeding on the infected person. This is microvilli carriers. 10 to 14 days. Mainly, microvilli mosquito develop in the 3 stages under the optimum conditions, temperature and humidity, duration of the life cycle, mosquito extensive incubation period is 10 to 40 days. The adult worm survive 5 to 8 years in a lymphatic system. Someone long. As the record as a 2022, 5.5 lakh lymphedema and 1.5 lakh hydrocele cases reported till 2022. The northwestern state union territory, namely Jammu Kashmir, Himachal Pradesh, Punjab, Haryana, Chandigarh, Rajasthan, Delhi, Uttaranchal or Uttarakhand, northeast state, namely Sikkim, Arunachal Pradesh, Nagaland, Meghalaya, Mizoram, Manipur, and Tripura, are known to be free from the indigenous acquired filariasis infection. The cases of filariasis have been recorded from Andhra Pradesh, Assam, Bihar, and many more like Uttarakhand, West Bengal, Pondicherry, Andaman, Nicobar, Daman, Diu, and Dadra Nagar Haveli and Lakshadweep. And National Filariasis Control Board are after a pilot project in Orissa, 1949 to 1954, National Filariasis Control Boards were launched in the country in 1995 with the objective of limiting the problem to undertake control measures in endemic area and the train personals to main the program. The main control measures were mass DEC administration, antiviral measures in urban area, and indoor residual spray in the rural area. NFC strategies, uh, recurrent anti-larval measures at weekly intervals, environmental methods including source reduction by filling detached pitch, low-lying areas, dwelling, distinct, etc. Biological control of mosquito breeding through larvivorous phases, anti-parasitic measures through detection and treatment, microvilli carriers and disease, person with the DEC by filariasis clinic in town covered under the program. Indigenous cases have been reported from about 339 districts in 20 states or union territories. The endemicity rates of the states in Bihar, 22% of the cases are reported in Kerala, 20%, Uttar Pradesh, 19%, and Andhra Pradesh 13%, Tamil Nadu 13%, Goa 1%, 2% in Lakshadweep, 4% in Madhya Pradesh, and 6% in Assam. The figure shows that all the districts in indigenous reported elimination of lymphatic filariasis. Many programs have launched for the elimination of the lymphatic filariasis. One is the two-pillar strategies like mass drug administration. Mass drug administrations from 2004, initially with the single dose DEC, and in 2007 with DEC plus albendazole co-administration. This is the two pillars mass program. So, uh, 2019, 87.2% to regionals mass drug administration was provided to the for the elimination of the lymphatic filariasis in many districts. The triple therapy is very much important. This is launched in 2028. First of all, is coverage in Arwal district, Bihar. After that, the triple dose therapy was covered 2019, and many districts Arwal, Allahabad, Chandoli, Hardoi, Lakhimpur Kheri in Uttar Pradesh, Unnao, Sitapur, Varanasi. These are the main districts of the some states which provide a triple drug therapy. Most of the cases come from to Lakhimpur Kheri, and many triple drug therapy has been given 95%, 74%. The conclusion is the agency and complexity. Filariasis in India poses a persistent public health challenges, simply the urgent and complex nature of interventions, long-standing efforts. First up, the National Parallel Control Program in 1955. Current scenario reveals resources challenge in disease control, magnitude of effect, and estimate 31 million individuals with microfilariae ends, 23 million symptomatic cases in highlight the substantial impact of the public health. A staggering 473 million individuals remain susceptible to infection, underscoring the need to comprehensive intervention strategies. Bacteria bronchitis transmitted by Quincy fasciitis accounts 99.4% of filariasis burden in India. So nocturnal priority and global burden is India contribute the 41% of worldwide lymphatic filariasis cases, reflecting the global significance of issues. Regional endemicities like Bihar, Kerala, Uttar Pradesh lead to endemicity rates, while states in Goa, Lakshadweep show lower rates, indicating regional variation. MDA pilot trials, ongoing master administration trials in 7 states underscore the critical need to targeted intervention addressing 86% of microfilarial carriers and 97% of disease cases. Multi-pronged strategy addressing filariasis depends on a multi-pronged strategy involving collaboration, innovation, sustainable public engagement, building on historical efforts, and interacting new insight is crucial for the future success. So this is the my review on the accessing the contemporary landscape of lymphatic filariasis in India. Thank you so much.
Thank you so much. Now next participant is Komal Sahu. Komal Sahu.
Yes, sir.
Yeah, please start.
Dr. Sushil Kumar.
Is this visible?
No, sir, not yet.
And now?
No, sir, I can't see my slides.
Now?
Yes, sir. It is
Okay,
visible,
please
sir.
start.
Thank you, sir. Good morning, everyone. Myself, Komal, from Maharishi Markandeeshwar University. I'm a PhD scholar and doing my PhD under the supervision of Dr. Raj Singh. And today I'm presenting on topic biological control of Parthenium hysterophorus, a noxious weed, a threat to human, animal health, and environment. So please, next slide. As everyone is aware of Parthenium hysterophorus, it is a very common noxious weed which can be found everywhere in India and Australia and parts of Africa. This is the scientific classification of Parthenium hysterophorus. It belongs to the order Asteriales, family Asteraceae, genus Parthenium, and species Hysterophorus, and we called it as Parthenium hysterophorus. This is a very harmful noxious weed which is considered as one of the worst weed ever discovered and is commonly known as carrot weed, white top, Chatak Chandni, Congress grass, Gajar grass in different parts of India. The weed has now spread to around 35 million hectares of land in India since it was first discovered in 1955. Moving on to the next slide. So let's start with a basic introduction. Parthenium hysterophorus is a common noxious weed which belongs to the Asteraceae family and causes several economic losses like health problems, habitat destruction in many tropical and subtropical countries. The weed came into India accidentally as seed contaminant with wheat. The Parthenium was first discovered in Arunachal Pradesh and Nagaland in 1810, and its presence was first registered in Pune in 1955. Parthenium weed develops very quickly, creating an adult plant into 1.5-meter tall and produces flowers early. A single flower produces 5 seeds and it produces around 15,000 seeds in its lifetime. There is a very harmful sesquiterpene lactone found in Parthenium hysterophorus, namely parthenin, which is harmful to livestock as well as animals also and poses as serious health risk to the people. For example, it causes dermatitis and skin rashes in humans as well as in animals, emaciation, tissue rupturing, internal organ hemorrhages, and death are the hazardous side effects of this harmful weed. Next slide. The next slide. Parthenium hysterophorus is very harmful to human beings as well as animals also, that it can also invade all kind of crops and herbaceous plants. It may have an impact on natural diversity and lead to the extinction of local flora if left unchecked. Parthenium is now a major problem throughout all of the India, where it has spread to neighboring countries by cars or contaminant of seeds that have been shipped. So it is very important to control this harmful weed. The control of Parthenium weed is a very difficult task due to its ferociously growing nature. Many studies are being conducted to determine the most effective and affordable method to control this weed around the world, which include physical, chemical, and biological control. The study shows that the biological control is a very suitable method to control this harmful weed because of its affordability, environmental safety, and sustainability could be a key component to control this harmful weed. Moving on to the next slide. So this is the list which shows the status of Parthenium hysterophorus in different states of India. This is the overall spread and infestation level which shows the high, medium or low level of the Parthenium hysterophorus in different states. Next slide. This is the life cycle of Parthenium hysterophorus. A mature Parthenium weed starts flowering after 1 month and it continues up to 3 months. After that, the seeds are produced. A single plant produces around 15,000 seeds and 624 million pollens throughout its lifetime. After that, the seeds are dispersed through grains, insects, animals, and vehicles, and then seeds germinate within a week, converting it into a mature Parthenium weed. The whole life cycle of Parthenium hysterophorus takes up to 90 to 120 days to complete. Next slide, please. So these are the harmful effects of Parthenium hysterophorus. The Parthenium hysterophorus weed is very harmful. There are many harmful effects, which are some are mentioned here. For example, Parthenium hysterophorus causes asthma, bronchitis, hay fever in humans, and dermatitis in humans, cattle, and domestic animals. It infects pastures and farmland, causing often disastrous loss of yield. The weed is also responsible for bitter milk disease in livestock as it taints meat and milk. These are the pictures which shows the harmful effects of Parthenium. The first picture shows the skin allergies in humans. It also causes skin allergies in animals also. It reduces the crop yield and it taints the meat and milk, and it also competes with the pasture species. Next slide. So next slide, please. Please change the slide. So now what is the biological control? Biological control is the intentional use of a weed's natural enemies to reduce its number. This is known as the biological control. Arthropods like insects, mites, and their relatives, bacteria or fungi are some of weed's natural enemies. These control agents prevent weed growth, reproduction, and spread by eating the weed or inflicting disease on it. Biological control of Parthenium is the most cost-effective, environmentally safe, and ecological viable method. So we may prefer this biological control. Different methods for biological control of Parthenium are used, for example, stem-carrying moths, leaf-eating beetles, stem-boring weevils, and various fungal isolates have been used globally with varying degrees of effectiveness. For example, Gyrogamma biocolorata, a leaf-eating beetle, was also identified as biocontrol agent which controlled the Parthenium by feeding on it. A rust pathogen, namely Puccinia abrupta and Puccinia malampodi, which is capable of controlling this weed also. Next slide, please, sir. Biological control of Parthenium mysteriforas is categorized into three categories. The first one is inundative biological control, which involves an agent that occurs as a pest in its native region, is cultivated, mass-produced, registered, marketed, and then spread inundatively like chemical pesticides, for example, Fusarium pallidorosium. In the classical biological control, the release of coivoid pathogen into an exotic environment where the target is an alien, for example, Gyrogramma bicolorata and Puccinia abrupta. The third one is integrated weed management, which involves a collection of diverse tactics with the shared goal of efficiently managing invasive weeds, for example, Astravelas grossa and Clitoria ternatia along the Gyrogramma bicolorata and Epiglemus tenuana. Next slide, please, sir. Now, what are the biological control agents and what are the application? Why we use biological control agents? Control of plant pathogens and diseases caused by them through antagonistic microorganisms is termed as biological control agents. Insects, fungi, nematodes, snails, slugs, competitive plants, and microorganisms may be the biological control agents of the Parthenium. Some examples of biological control agents of Parthenium mysteriforas are Gyrogramma bicolorata, Listonotus cytosiphanes, Epiglemus tenuana, Cormenta itheki, Puccinia abrupta, and Puccinia malampodi. We use biological control agents as they are non-toxic to human and animals. They are low cost, host specific, and not polluted also. Moving on to the next slide, please. So these are the pictures of some biological control agents of the Parthenium mysteriforas. The first one is Gyrogramma bicolorata, which is also called as leaf-eating beetle. The second was is Listonotus cytosiphanes, called as stem-boring weevil. The third one is Epiglemus tenuana, called as stem-bearing moth. And the last one is Cormenta itheki, called as root-boring moth. Next slide, please. These are the two rust pathogens, which are also considered as the fungal pathogens, which are also considered as the biocontrol agents of the Parthenium mysteriforas and shows good results in controlling this harmful weed. The first one is Puccinia abrupta, variant Parthenicola, which is called as winter rust. And the second one is Puccinia malampodi, called as summer rust. Please, next slide. So these are the list of some fungal pathogens which are found on the Parthenium mysteriforas or isolated from the Parthenium mysteriforas. So we can use these fungal isolates as biological control agents to control this harmful weed. Next slide, sir. These are all fungal isolates. Please skip. So let's conclude. Parthenium hysterophorus is mostly recognized for its effects on the natural environment, negative health effects on humans and animals, and disturbance of the local native plants community habitat. It can outcompete palatable plants that are beneficial to livestock. Therefore, it is imperative to eradicate this dangerous weed. Although various tools have been proposed in recent decades to suppress this harmful weed, but biological control presents an economical, secure, and long-term solution to control this invasive weed. So it is very crucial to take into account in additional biological control agents to control this harmful weed. Thank you. Thank you so much, sir, for presenting my slides.
Yes, thank you so much.
Thank you so much, sir.
Now the next participant is Suruchi Kumari, BVAU.
Yes, sir.
Yes, please present.
So is it visible?
No, it's not visible.
So now?
Yeah, it's very much.
Okay. So good morning, everyone. Myself, Suruchi Kumari. I'm from Babasaheb Bhimrao Ambedkar University. And my topic for presentation is novel antimicrobial peptide from herbal formulation. So antimicrobial peptides got into the picture when there comes the antibiotic resistance. And antimicrobial peptides, they are biologically active molecules produced by the wide variety of organisms as an essential component of their innate immune response. So these act as a bridge for innate or acquired immune system, and their primary role include the host defense by exerting the cytotoxicity on the invading pathogenic microorganism. Antimicrobial peptides can be best suitable as the drug candidate because of their broad range of activity, less toxicity, and decreased resistance development by the target cell. They are very specific and it depends upon the interaction of peptides with the microbial cells, which enable them to kill the specific target cell without affecting the host cells. And net charge and hydrophobicity also play a crucial role in the cellular association of these peptides by selecting the target cellular association of these peptides and selective target cellular membrane in exerting the antimicrobial activity. So these are the some of the antimicrobial peptides with their structure, and they are found to exist in a wide range of secondary structures such as alpha helices and beta strand with one or more disulfide bridges, loop, and extended structures. Extraction of the antimicrobial peptides, first we have to select the plant material because antimicrobial peptide from plant can be extracted from seed, leaves, roots. So first is the selection of plant material, then it is crushed and blended. The third process is the frozen. It is frozen and thawed after centrifugation. Well, whatever the residue is collected, that is discarded. The supernatant which is collected is allowed to precipitate in the ammonium sulfate. Then again we go for the centrifugation, and after collecting the pellet, we dissolve it in the deionized water, and again we go for the dialysis. And in the gel filtration chromatography, it is also known as the size exclusion chromatography, where we separate the molecule on the basis of the size. After that, go for the fraction is collected, and again we go for the dialysis. Then the purified antimicrobial peptide is collected, and we get the amino acid sequence. Now, if we talk about the mode of the action, then there are three major mode of action which is known. First is the barrel-Steve model. It is energy independent mechanism. So peptide monomer get aggregated on the surface of the membrane, and the non-polar side chain direct towards the hydrophobic lipid cell membrane, and the hydrophilic surface of peptide point inward and forms the water-filled membrane, and that releases the intracellular content of the cell. The second one is the carpet model. It is also the energy independent mechanism. In this, peptide associate at the surface of the cell membrane, and when a threshold concentration is reached, they induce the membrane permeation and lead to the disruption of the cell and also causes the cell death. The third is the macropinocytosis, and it is energy dependent process. In this, plasma membrane act on the plasma membrane of target cell get folded along with the peptide, and it form a vesicle which is known as the micropinosome, and the antimicrobial peptide within the vesicle get released into the cytoplasm and it exert the antimicrobial activity. Defensins comes as a major antimicrobial peptide, and it has been reported for many crops of economic importance which aim of increasing resistance to phytopathogens. The antimicrobial peptide which is extracted from the one plant, which is the Redis defensin, which is known as RSAFP2. It is the first heterologous plant peptide expressed in the another plant. So it is conferring the protection against the fungus. The other plant antimicrobial peptide with higher potential use have also been used to transform the different plants, like the MSDEFL alpha alpha defensin, which is expressed in vivo in tomato, and it provides protection against the fungi if oxysporum. Now the current and the future research aspects are basically there are three main areas. First is to control the expression of the AMPs so that they occur in the presence of pathogens
Good
and
morning, sir.
preferably at
सर अभी आप आएंगे ना आधे घंटे में?
attacked sites.
जी जी जी. अच्छा आप ऑनलाइन ही है. ओके 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 सर.
Second is the improvement of desirable characteristics such as high activity and low cytotoxicity by some modification. And third is the synergic expression of different AMPs with diverse mechanism of action. These are my references. Thank you.
Okay, thank you so much. Now our two oral presentation are left. After that, the session will be start for the lecture invite guests. Next one is Supriya Upadhyay. After that, Avilasa Sahu, and then invite guest speakers. Lecture will be start. Miss Supriya Upadhyay.
Yes, sir.
Yes, please.
मम्मी को शेयर करना है इसका.
Suruchi, can you please share the slides?
Yeah, just wait a minute.
Good morning, everyone. I am Supriya Upadhyay, currently pursuing Masters in Zoology. My topic for the presentation is enzyme targeting for the development of novel antimicrobes. My content for the presentations are introduction and various enzymes that are being used in the development of antimicrobes. As we know that we are living in a world of various diseases where the bacteria and viruses are getting resistant to various antimicrobes, and so we need to develop various antimicrobials to cope up with them. So AMEs are one of the targets to do that. AMEs are a group of enzymes that can directly attack bacteria, interfere with their biofilm formation and catalyze antibody producing reactions. It is of course because of the development of the antibiotic problems and the loss of effectiveness of the various antimicrobes that we are using today. Um one of the main action of the enzymes that we are doing is the QS signaling molecules, that what they do is they transfer the bacteria to the status of high resistant to the effects of antimicrobial agents. That is the biggest problem right now. In addition, the action of enzymes themselves and in combination with other biocides, we are doing this. We are using enzymes with other products also to create various antimicrobials. Next. The first of them is oxidoreductases. And in oxidoreductases also, we will talk about glucose oxidase that are very much effective against Pseudomonas aeruginosa, Escherichia coli, and Staphylococcus aureus. They are very much effective because of their ability to inhibit the growth of certain bacteria using because they can lower the pH of culture medium with the help of hydrogen peroxide and gluconic acid. Next. Next is carbohydrases. Carbohydrate eradication of bacterial biofilms remains a major challenge, which is because they become resistant. So our current strategy is to obtain antibacterial and antiviral activity from those enzymes and hydrolyze the peptidoglycan which is present in their bacterial wall. Lysozyme is one of the hydrolase that catalyzes beta 1,4 glycosidic bonds. The lysozymes are very popular in the product functionalization because of their thermal stability at different temperatures and the antimicrobial activity against various bacteria and fungi. Next. We will specifically be talking about lysozyme. The antibacterial activity of lysozyme immobilized on cellulose nanofibers was studied, and I took it from there. These antibacterial products proved to have good properties as the enzyme activity decreased by 12 to 30% during immobilization, and after 9 cycle duty, after immobilization, the antibacterial activity of lysozymes was reduced. Lysozyme has been proved to have the greatest antibacterial activity against black yeast and salmonella that are one of the biggest threats right now in the world. Next. The effects of the combination of antibiotics and enzymes on Escherichia coli cells were more pronounced than the effects of antibiotics and enzymes alone, especially at the pH of 5.7 and 8.0. However, this effect is weaker for L monocytogenes. The activity of Salmonella typhimurium was better at pH 5.5 and 6.0. So Salmonella has been the course of study for the lysozymes in this. Next. Another enzyme that we use is amylase and pectinases. Biofilms that are formed with the polysaccharides have strong populations of cell in which bacteria are highly resistant to antibiotic action. That is why they become resistant to many of the antimicrobials. One of the most effective methods of biofilm removal is the disruption of biofilms using enzymes that hydrolyze and the polysaccharide material and weaken the biofilm matrix, which is one of the main strategies to create antimicrobials. Enzymes such as glycoside hydrolases are the enzymes that are being used to create such antimicrobials with highest antibacterial properties. The operation of materials involves the immobilization of enzymes on their surfaces in the formation of chemical interactions between enzymes and supports that ensure the maintenance of high catalytic activity of enzymes. Next. Alpha amylase has been proved as one of the most important antimicrobial creating enzymes, as they create one of the best antimicrobials when combined with other enzymes. And synthesis of alpha amylase from bacillus was proved to have to give that effect. The use of proteases derived from type 1 bovine pancreas is known to prevent bacterial biofilm formation, which is our main target. The combination of enzymes enabled E. coli and S. aureus to inhibit biofilm formation by 78% and 90% respectively. The antimicrobial film materials that reduce biofilm formation have been developed, and alpha amylase and lysozyme were covalently immobilized as antibacterial enzymes on polydopamine chloride functionalized polyether sulfone membranes. So this leads to the formation of biocompatible antibacterial surface. Also, this multi-layer protection prevents membrane fouling and biofilm formation. To prevent biofilm formation is also very important to create reduce the resistance of antimicrobials. The result of microtiter test and flow cytometry showed the structure of such material ensured the removal of more than 87% of biofilms. Next. Therefore, there's an interest in pectinases as enzymes that can produce antibacterial compounds for studies of different products conducted as interesting studies. Also, the pectinases and alpha amylases uh, have been proved from this paper that they have antibacterial properties and can be used with other biocides and other enzymes to remove biofilms and prevent bacteria from adhering to the treated areas. Next. Next are proteolytic enzymes. In bacterial biofilm, in addition to polysaccharides, cells are also surrounded by extracellular proteins, nucleic acids, and lipids secreted by cells. The drugs contain composition of different organisms, so they are used to reduce the risk of contamination associated with biofilm formation. Enzyme belong to the class hydrolysis that remove peptide bonds of amino acids and proteins and peptides and are divided into exo and endopeptidases. Proteolytic enzymes are originally derived from plants, animals, and microbial organisms and are widely used in pharmaceutical, food, and nutrition industries. Proteases have been studied for their antibacterial film forming ability. Using alpha, chemotrypsin, and physin as examples, studies have shown that in addition to the hydrolysis of proteins involved in biofilm formation, they can disrupt the cell communication due to cleavage of signal peptidase type 1. Next. Chemotrypsin and physin are also used for making various antibiotics and also solely and also with
Please
the
bind up.
Yes, sir. Yes, so this was the last actually. Okay. Conclusion. We use various antimicrobial properties for making them useful in the current world. Okay. Thank you.
Thank you so much. Due to the time, 10 o'clock, now we are shift our next program, that is invited guest lecture. On the behalf of the organizing committee and my own behalf, I would like to invite our today's invite speaker and eminent academician, Dr. Reena V. Saini. She is currently member secretary, Institutional Biosafety Committee, MMDU, and professor, Department of Bioscience and Technology, MMDU. She has excellence in innovation strategy for the national building and mankind, She has huge contribution in science and academia. She published about 106 research papers and hundreds of patents, among them dozens of the patents got awarded by the IP India. By these words, we welcome Professor Reena V. Saini, ma'am. Please, ma'am.
Hello, am I audible?
Yes, ma'am. Audible, very much audible.
Okay, thank you so much. I'm really humbled with this wonderful introduction. And I also want to thank International Association of Zoologists and Siddhartha Group for inviting me for giving this talk, and congratulate all the organizers for organizing this wonderful international conference. Okay, so I'm sharing my presentation.
Yes,
Is it
ma'am.
visible?
Yes, ma'am, visible.
Okay, so the topic of my today's presentation is granulysin as a biomarker for activated lymphocyte to assess the efficacy of phytocompounds as cancer immunotherapeutics. So my lab actually work on cancer biology and basically on cancer immunotherapeutic agents. And these agents are plant-based agents, and we also work on nanotechnology. So on today's talk, I'm going to talk about cancer immunotherapy, and I'm going to talk about plant compounds, how they can be used to activate human lymphocytes. So a little introduction about the cancer. Cancer is actually a condition in which your body cells grow in an uncontrolled manner. And these cells lost their property of cell growth, as well as they lost their property to undergo cell death. And in most of the cases, this is caused by oncogenes, the mutations in the oncogenes, tumor suppressor gene, and DNA repair genes actually cause cancer in these cases. Cancer is really a global burden, and in this slide you can see that in Asia, like 48% cancer incidences are present in Asia, and India is a part of Asia, so that is why I'm showing this. And in India, every year, like 11 lakh cancer patients are registered, and around 7.8 cancer related deaths happen in India every year. So for the cancer, there are many therapies available. For example, hormone therapy, surgery, radiotherapy, targeted therapy, chemotherapy. All these therapies are available, and many cancer patients are getting cured. But still, the cancer milieu is different for every patient, and every patient has a different body system and immune system. So there's a still need for new therapies. This could be personalized therapies. This could be more advanced one, which lesser side effects. So as my lab work on cancer immunotherapy, so my talk will be focused on this immunotherapeutic part. So immune response in case of cancer is mainly mediated by cytotoxic T lymphocytes and natural killer cells. These cells have granules which release their content in the vicinity of cancer cell and then induce cancer killing pathways in the cancer cell. The most important protein which I have worked during my postdoc is a granulysin protein. This protein is present in both NK cell and cytotoxic T lymphocyte granules. The important part about this protein is that this is only expressed when these cells are activated. In the naive cells or in non-activated cells, these granulysin protein expression is really, really low. But when they get activated, the expression of granulysin get enhanced. This is the work which we have published in like 2011 way back, where we show that granulysin can enter into the cancer cell with the help of perforin or could be without perforin. And it can activate either mitochondrial cell death pathways, and when it enters through perforin mediated pathway, it activated endoplasmic reticulum apoptotic pathways in the cancer cells. And also in New England Journal of Medicine, it has been shown that in the patients where cancer metastasis is very less, the granulysin expression is really high. So there is an indirect correlation. And on the basis of this paper, it was elucidated that granulysin can act as a immunomarker for these cancer patients. That means in patient where granulysin expression is high, their prognosis will be
Excuse me,
better.
ma'am.
Yes, sir. Yes, sir.
मैम एक्चुअली स्लाइड शेयर नहीं हो रही है शायद, मतलब सिर्फ फर्स्ट स्लाइड में ही है.
Okay, okay, okay. Let me now.
Yes, ma'am. Yes, ma'am.
Yes, okay, sorry about that. So I'll not go to the full screen. Then I'll show this in this mode only. Okay.
Okay, ma'am. Okay.
Okay. So we found that when a patient has a metastasis and a very third or fourth stage cancer, then granulysin expression is very, very low. But when cancer has been controlled, the granulysin expression is really high. So on basis of these paper, and there are several other reports, it was concluded that granulysin can actually act as a immunomarker. So, because my talk is based on immunotherapy, we're going to talk about plant-based immunomodulators and how granulysin is correlated with them. These immunomodulators can be immunostimulant or these can be immunosuppressant. Okay, before going further, can I ask, can you see this slide? Is this slide visible?
Yes, ma'am. Yes, ma'am.
Okay. So the plant immunomodulators that I'm going to talk today is the Datura stramonium. You might have heard about Datura. This is a plant which actually causes hallucination because of presence of the alkaloids. There is a high content of alkaloids, but this plant also contains very high content of non-alkaloids. And these non-alkaloids have been shown to act as immunostimulators. The plant called as Videnia somnifera. In this plant also a lot of non-alkaloid content is there, and जिसको हम अश्वगंधा कहते हैं उसमें भी बहुत ज्यादा non-alkaloid है, and that is why it is a very good immunostimulator. So this study start with this plant where we actually use this plant leaves and then we isolated non-alkaloids, which then we checked on human peripheral blood mononuclear cells, in short human lymphocytes, and then we checked their immunomodulatory potential. So the hypothesis of this work is whether the turastramonium immunomodulators or the isolated compounds can act as immunostimulators, and how the granulysin can act as a biomarker to understand the effectiveness of any tumor immunotherapy. This work was granted by Department of Biotechnology, and this was a scheme of pilot project grant for young investigators in cancer biology. And I received like 25 lakh grant for this work. So to start with, we took the turastramonium leaves, and then from this leaf, after a long fractionation and chromatography, and then identification by NMR, characterization by NMR, we were able to isolate 3 molecules. I'm not going into the details of isolation and characterization of molecules. I will directly go to the immunotherapeutic part. So we actually get 3 molecules, the turalactone, deoxyvidestremolite, and the turilin. The turalactone, D1 and D23 that we have mentioned in our further slides, these were already seen in the turastramonium, but the turilin was the first time isolated from the turastramonium leaves in our study. So this is the plan how we fractionated and did column chromatography and did the NMR studies to identify these molecules. This is the structure of these 3 molecules. I'll not go into the detail. I'll rather go into the immunomodulatory activity. So this is the in vitro assay where we isolated human peripheral blood mononuclear cells. These are human immune cells which are comprised of T lymphocytes and natural killer cells. And when we incubated these human lymphocytes with these compounds D1, D23, and D27, you can see increase in their proliferation. The proliferation was assessed by MTT assay. And with the increase in concentration, there's increase in the proliferation. Proliferation is the first indication that these compounds are able to work on these cells. And once these cells are proliferated, then we'll look for the activation markers. This is again the microscopic study where you can see the unactivated cells and then cells activated by D1, D23, and D27. You can clearly see lot many cells, and you can clearly see the size of the cell. The increase in the size also tell that there is an activation. The effective concentration was like 1 micromolar for all three of these molecules. Then we looked at the another immunomarkers. As immune cells release cytokines, IL-2, interferon gamma, and TNF alpha are the cytokines which actually are released by the immune cells in their neighboring communities, so that activation of total immune system or activation of immune response take place. We looked at the interleukin 2, interferon gamma, and TNF alpha after 24 and 40 hours of treatment of the immune cells. You can see that these molecules can activate interleukin 2 secretion, interferon gamma secretion, and TNF alpha secretion after treatment with these molecules. This is the flow cytometric data where we looked at the CD3, CD8, and CD56 marker. I want to tell you, audience, here that CD3 and CD8 are T lymphocyte markers, and CD56 is the natural killer cell marker. And you can see the increase in these CD56 marker and CD8 marker after treatment with D1, D23, D27 molecules after 48 hours of incubation. So as we have told earlier that we want to correlate this data with the granulysin presence or the expression of granulysin. So this is a very nice picture which showing that cytotoxic T lymphocytes is actually interacting with cancer cells and they are releasing their granules so that cancer cell killing can be enhanced. So we want to check whether granulysin expression is actually increased after treatment with these molecules. So we took the immune cells, human immune cells, human lymphocytes. We treated them with these compounds D23, D1, D23, D27. And this is the flow cytometric data which showed the expression of granulysin. You can see there's an enhanced expression of granulysin as compared to unactivated cells. In the unactivated cell, there's hardly any expression, whereas there is like 39%, 35%, and 63% enhancement in the granulysin expression after treatment with these molecules. This is the time graph which shows that with the increase in the time from 24 to 48 hours, granulysin expression goes up and then slowly it goes down up to 96 hours. Then we did a very interesting co-culture assay where we actually incubated these activated immune cells with the cancer cells. So just in brief to tell you, what we did is we took these immune cells, we incubated them with the D1, D23, D27 molecules separately, and then we co-cultured them with cancer cells. These pictures are of the co-culture assay. And you can see that in the presence of D1, D23, D27, you can hardly see any cancer cells because cancer cells are undergoing cell death, and then there is a apoptosis and cell breakdown. So that is why very less cancer cells are visible in this picture. And when you quantitate this, the cancer cell used in this assay is actually colon cancer cell lines. We use CT116, SW620, and one pancreatic cell line, which is PANC1. And with the increase in effector is to target ratio, the effector are activated PBMCs, immune cells, and the target cells are the cancer cells. You can see with the increase in effector is to target ratio, there's increase in the cancer cell death of both colon cancer cells and the pancreatic cancer cells. So this study actually, the take-home message is Datura lectins or the molecules isolated from Datura can activate human immune cells under in vitro conditions. They are able to release cytokine, they are able to release granulysin and activate these CD3, CD8 markers. Once activated, they are able to kill cancer cell very effectively. That leads to killing of cancer cell or apoptosis of the cancer cell. We also did the mechanistic studies later on, but I'm not going into the detail of mechanistic study. But we found that these activated lymphocytes or immune cells have enhanced reactive oxygen species generation and enhanced mitochondrial activation. So to take home messages is actually granulysin can act as an immunomarkers in these activated human lymphocytes for their immunostimulatory potential. This work was published in Phytomedicine Journal and Minerva Biotechnology and Biomolecular Research Journal. And we also able to fetch 3 patents out of this research work. So this was a very nice work done one of my PhD scholar Gaurav Chandan. Currently Gaurav is working as a postdoc in UCLA in USA. So that's all for today's talk. Thank you so much for giving me this opportunity. Now I can take questions, if any. Thank you.
If anyone asks to any question, please raise your hand or ask the questions directly.
Thank you so much, ma'am, for nice deliberation and informative talk. You enlightened us in a very fascinating manner with mechanistic approach in the cancer biology, treatments, strategies. Really, you are doing an incredible job at the institutions, as you have many more assignments. So you have shared some time with us from your busy schedule. We are highly appreciative for you as well. So with these words, I would like to extend my congratulations, and it is a great opportunity to honor you with a token of memory from my society, means from the International Association of Geologists and from my own behalf. So please accept my honor with in the form of token of memory as Innovative Scientist Award from International Association of Geologists.
Thank you so much, sir. I am really honored and humbled. Thank you, sir.
So congratulations, ma'am.
Thank you so much, sir. Thank you so much. Okay, sir. Thank you so much.
Thank you, ma'am. Thank you. Now over to Siddharth, next proceeding.
Thank you so much, ma'am. It's a very fruitful lecture about cancer. Now I would like to invite Ms. Diksha Sharma to invite Dr. Safakatullah Nannu sir. Please hand over to Diksha Sharma.
Good morning, all. I would like to introduce Dr. Shafaqatullah Nanu. He is a dedicated and diligent professional with a wealth of experience in academia and industry. He is currently working as an assistant professor at Mangalore University in Dakshina Kannada, which is located in the state of Karnataka, India. Dr. Shafaqatullah is an expert in the fields of biotechnology, molecular biology, plant biotechnology, animal biotechnology, microbiology, nanotechnology, and biochemistry. He has 8 years of experience across his research and teaching jobs, so he knows a lot about the subject. He has been active in the field of education since July 2016, when he began working as an assistant professor at Mangalore University. He has been a teacher for 7 years and 5 months of total. Dr. Shafaqatullah has extensive knowledge in the scientific field because of his time spent as a senior scientist fellow between August 11 and June 2016. During this time, he did research in the lab and went out into the field to collect the data. Prior to beginning his current position as Senior Research Fellow at Mangalore University in June 2011, Dr. Shafaqatullah worked there as a Senior Research Fellow from February 2010 to June 2011. During this time, cancer and probiotics were at the center of his research. He also explored freshwater mussels in his studies. Dr. Shafaqatullah went to Bangalore University between 23 to 2005 to earn his master's in biotechnology. He also did a specific study on the link between probiotics and cancer between 2010 and 2015. Dr. Shafaqatullah's wealth of experience and expertise in variety of biotechnology related fields is indicative of his commitment to both research and teaching in this field. Over to you, sir.
Yes, thank you, madam, for your elaborative introduction. So now I'm going to present my topic on probiotics. So let me share the is it visible? Is it visible?
Yes, sir, it's visible.
Yeah, okay. So good morning. So myself, Dr. Nannu Shafaqatullah from Mangalore University. So today I am here to present a research topic on probiotics from non-fermented food sources. So generally these probiotic organisms, so usually we are isolating from the fermented food products. So but what we have decided is to identify and isolate this organism from the non-fermented food products. So probiotics, these are live microorganism which when we administer or when we intake, they are having a good effect on the health of any individual. And these probiotics include especially the genus of Lactobacillus, Lactococcus, Bifidobacteria, Pediococcus, Streptococcus, Leuconostoc, etc. And when you take their a mechanism of action, so they play very much important role in our whole GI tract, especially in the small intestine. So they have adhesion to the epithelial receptor. So when they are attaching to the epithelial receptor, we can prevent the attachment of pathogenic organism on the intestine. So the pathogenic organism from the intestine can be excluded. And they have the property of production of inhibitory substances. They are responsible for the production of this one lactic acid. They're responsible for the production of hydrogen peroxide and other secondary metabolites. So because of these secondary metabolites, some of the secondary metabolites acting as the inhibitory compounds for the pathogenic organism which are present in the intestine. They are working as the competitors for the nutrition in the intestinal tract. So there will be the competition between this probiotic organism as well as the pathogenic organism in the intestine. So because of this high competition, the pathogenic organism can be easily eliminated from the system. They have the aggregation capacity. So in the intestine, they coagulate or they aggregate with the pathogenic organism. And because of this aggregated complex which is forming between the pathogenic organism and the probiotic organism, along with the probiotic organism, this pathogenic organism which are present in the small intestine can be removed out from the system. They have all this very much effect, very good effect on the immune system. They are helping for the immune modulation also. So overall we can say that these probiotics are giving very much good effect on the system. And the aim behind this probiotic isolation from non-fermented food product is to find out their effect on the colorectal cancer. So that was the main objective of our research, to find out whether these probiotics which were isolating from the non-fermented food, they got any effect on the colorectal cancer treatment. So what we did is we isolated this probiotic organism, that is a lactic acid bacteria, from around 15 probiotic organisms. This one, 15 non-fermented food products, that is we isolated from the cow milk, buffalo milk, goat milk, sheep milk, cucumber, cabbage, etc. So these are the different non-fermented food products we have selected for this purpose. So what we did is the simple isolation procedure we have followed, that is a morphological character, that is based on their morphology we identified, that is the initial stage of identification in that morphology. The colony morphology, simple staining, motility, endospore formation, and growth in different broth as well as land has been observed. Then we went on for the biochemical identification. So the only the morphological character will not help you to identify the probiotic organism or the lactic acid bacteria. So we went on for the biochemical characteristics. So in that biochemical characteristics, we did almost all the general biochemical characteristics to differentiate between the different organism, and especially we have done the carbohydrate fermentation test. So this carbohydrate fermentation test is very much important for distinguishing the organism between different genus based on their fermentation pattern, based on their uh, mechanism to utilize different carbohydrate. We can uh, identify some of the bacteria, not in the advanced stage but in the initial stages. Then we studied about their physiological characteristics of the isolate, because we want to see that this organism, whatever we are isolating and which we are calling as probiotic organism, whether we can use them in our food product, whether we can consume them, whether they can tolerate inside our intestinal tract or not. So for that we have done their growth in different sodium chloride concentration at different temperature we did to know whether this organism can be used in the storage of any food or food products. Then we have seen their effect in the different pH. So these are the results of our probiotic organism. So from these 15 sources, non-fermented food sources, we isolated around 55 different organisms, and we identified them from the morphological point of view, from the biochemical point of view, and finally we went to molecular identification. We have done the 16S rRNA sequencing. Then we identified around 22 organism out of this, because of the cost we could not do all the 55 organism. We have selected around 15 or 22 organism based on their different biochemical as well as physicochemical properties. Then we done the 16S rRNA sequencing, and we found out 22 different probiotic organisms. So these are the general morphological characteristics what we have studied, and this is their carbohydrate utilization pattern what we have studied in their identification process. These are their physiological characteristics of the isolates, that sodium chloride tolerance done by this organism. So when organism, they have seen that they can grow at a concentration of around 8 to 10 percentage of sodium chloride. So when you're using when the organism can survive in more concentration of sodium chloride, mean we can say that this organism can be used for the food products which contains more amount of the sodium chloride, maybe for example in the pickle or some other food product which contain more concentration of sodium chloride. There also we can use these organisms. Then you have done their pH as well as the temperature sensitivity. So this organism could survive at around 45-degree centigrade. So some few organisms, they have shown their growth even in the 50-degree centigrade, but the optimum temperature was around 35 to 37-degree centigrade, where we achieved a maximum growth in the organisms. Then we have seen their growth pattern to see in which way the organisms they are growing. So the maximum growth was around 3 days or 4 days. They have grown in a maximum. After that, fourth day of their this one multiplication, so their growth ceased. That means their death period started. And out of these 55 organisms, what we have done is we wanted to know whether all these organisms, whatever we isolated, they got the probiotic characteristics or not. So what we have done is we have done the screening test, in vitro screening test, to that is based on your ICMR, DBT as well as WHO. We have done 11 screening tests to identify the potential probiotic organism out of this 55. So what we have done is we have done the acid tolerance test, bile tolerance test, gastric juice tolerance, intestinal juice tolerance, autoaggregation, co-aggregation, bile salt, hydrolase activity, adhesion, hydrophobicity, antimicrobial activity, as well as a antibiotic sensitivity test. So these tests. If the organisms are passing these tests, means the organism can be called as the potential probiotic, because these are the uh, characters, or these are the criteria for their ultimate selection as a probiotic organism. In the sorry, so in the acid test, what I've done is we culture this organism at a pH of 1.5 to 3, for around 2 to 3 hours, because acid, we have to see that when we are consuming this organism, they first enter into the stomach. So the stomach pH will be around 1.2, 1.5 to 3 pH. So we have to see whether this organism can tolerate this acidic pH. So we have done this test by using all this organism, and this organism, they have shown very good tolerance to the acidic pH. So we can say that maximum organism, around 50 55 organism, more than 30-35 organism, they have shown good resistance to the acidic pH. So these are the graphs which shows their resistance to acid. And then bile. The next is bile, which is present in the this one, GI tract. This is the bile is also acting as an inhibitory compound for the microorganism. So we wanted to know whether this bile, which is present in our GI tract, whether it can prevent the growth of or survival of our probiotic organism. So to do that, we have done their survival in the presence of bile. We have seen their good survival. The survival rate was around 50 to 80 percentage of organism they could survive in the bile concentrations. And the bile concentration, we used three different concentration of bile we used. Then it is the bile which is present in our GI tract is around zero point 0.5, but we have done for 0.3 to 1 percentage of bile also we have done. But the organism, they have shown a good survival rate in the bile concentration. Then we have seen about the gastric juice tolerance, because when these organisms are present in the gastric environment, they should survive in that gastric, because it contains the large concentration of the pepsin. So we have seen whether this organism can survive in the maximum concentration of pepsin. So in the pepsin also, in the presence of pepsin also, we could see that the organism have shown around 30 to 35 percentage of survival rate we have seen by these isolated organisms. Then the tolerance to intestinal juice. In the intestinal juice, we are having the pancreatin especially. So we wanted to know whether this pancreatin got any inhibitory effect on this probiotic organisms. So, but we have seen in our study, we could see that the organism could show around 95 percentage of survival after 6 hours of growth in the pancreatin concentration. So that means we can say that these are the good organism which are having that special property which is preventing them from the effect of pancreatin. So they can easily pass from this pancreatin effect. Then we find out their auto aggregation. This because the auto aggregation plays very much important role for the survival of this organism inside the intestinal tract. So if the organisms are present as a single organism, they may be flushed out from the intestine. So they may not give a good effect in the intestinal tract. But when they are auto aggregating means self attachment of the organism. That is, each organism, they are forming clumps. So when they are forming clumps and attaching to the intestinal wall, they may not be easily eliminated or removed out from the system. So this auto aggregation, in the auto aggregation we have done about from 1 to 5 hours period, time period of auto aggregation we have seen. So the organism could have they have shown very good amount of auto aggregation property. That means they can easily stay in the intestine without getting eliminated from the intestinal flux. Then we seen their studied about their co-aggregation property. This co-aggregation help for the separation or elimination of the pathogenic organism. Autoaggregation is the self-aggregation. Co-aggregation is aggregation or attachment of this organism to the pathogenic organism. Maybe we got the pathogenic E. coli, or maybe we got the any pathogenic staph organism in the intestine, or Klebsiella or some other organism. So in this we have seen we have studied about their co-aggregation property with the staph aureus as well as the E. coli, and this organism they have shown good co-aggregation property. That means they can go and bind to the staphylococcus aureus, or they can go and bind to the E. coli. So they're forming the aggregate, and these aggregates can be eliminated from the system easily without getting a adverse effect by the E. coli or the staph aureus in the intestine. So we've seen around 76.24 or 34.11 percentage of co-aggregation property. Then we studied about their bile salt hydrolase activity, because this bile salt hydrolase activity of the organism plays very much important role in reducing the cholesterol of the system. So there is link between this bile salt hydrolase activity as well as the cholesterol. So the organism they have shown good bile salt hydrolase activity. So more the bile salt hydrolase activity means they got very good property to eliminate, or they got very good property to stop the synthesis of bad cholesterol in the body. So this 3 plus means more activity, and 1 plus means the less activity of bile salt hydrolase activity. So this is the figure which shows their bile salt activities. That is more the precipitation around the this one around the well means there is more amount of bile salt hydrolase activity by the organism. Then we have seen their studied about their adhesion property, because this adhesion property of this probiotic organism is helping to keep this organism intact, or the helping this probiotic organism to remain adhere to the intestinal tract for longer periods. So when they are remain attached to the intestinal tract for longer periods, so they will not give any space for the pathogenic organism, or they may easily eliminate the E. coli or Staph aureus or some other harmful organism. So when they are having more adhesion property means they will be attached to the intestinal epithelial cells. So the intestinal epithelial cell will not be harmed by some other pathogenic organism. So here you've seen good amount of adhesion property by this probiotic organism on the intestinal epithelial cells. So this is the protocol which we have followed to find out their effect or their adhesion on the CaCO2 cells. So more the adhesion means they can easily survive in the small intestine for longer period. Then we studied about their hydrophobicity property, and this hydrophobicity property is also helping for this organism to remain attached to the intestinal tract. So there is correlation between the adhesion as well as the hydrophobicity property. So in this hydrophobicity property, we've seen that this organism can they have around 70 to 75%, 70 to 75 percentage attachment property or the hydrophobicity property in the intestinal tract. So then we have seen about their antibiotic susceptibility of the isolate. So the need to find out their antibiotic susceptibility is when we are taking this generally what the doctors prescribe. They prescribe probiotic along with the antibiotics. So when we are intaking antibiotics, they are prescribing these probiotics also. So we wanted to find out whether the probiotics, whatever we are taking, it is getting affected by the intake of antibiotics or not. So we have used around 15 generally taken antibiotics to find out whether these antibiotics have any effect on the probiotic organisms or not. So if they are not having any effect on the probiotic organism, means these probiotics can easily survive even though we are intaking antibiotics. So, and this is the result of the antibiotic susceptibility test. So the majority of the organism, around out of this 55 organism, more than 20 organism, they were resistant to the antibiotics. So when they are resistant means we are intaking antibiotic maybe for some other diseases, but the probiotic which are present, which we are consuming along with these antibiotics, will not get affected. So we can call it as the resistant organism. Here we can call them as the potent or potential probiotic organisms. These are the slides which show their resistance against different probiotic organisms. Then we have studied about their antimicrobial activity. So we wanted to know whether this probiotic organism, they can produce some chemicals, maybe the bacteriocin, maybe niacin, maybe lactic acid, maybe hydrogen peroxide, or some other secondary metabolites. And these secondary metabolites are responsible to inhibit the pathogenic organism. So we have studied about their effect, the probiotic effect on different pathogenic organism. We used the Bacillus subtilis, we used E. coli, we used Klebsiella pneumonia, we used Staphylococcus, we used the fungus, that is the Aspergillus niger, we used albicans. So these are the pathogenic organisms, and we studied whether our probiotic organism, whether they can inhibit the growth of these pathogenic organisms or not. So in our study, we have seen a very good effect of our probiotic organism, direct probiotic effect on the pathogenic organism. So these 3 plus means they have shown a good inhibitory effect against the growth of these pathogenic organism. Only few organism they could not show the this one antimicrobial activity, especially on the fungus, but the majority of this bacteria, that is the pathogenic organism, they have been their growth has been hindered or inhibited by the presence of this probiotic organism. These are the results of that. Then we studied about the antimicrobial compounds which are produced by this probiotic organism. In that we have studied about the bacteriocin. We studied the concentration of bacteriocin, concentration of lactic acid, and hydrogen peroxide produced by this organism. So this bacteriocin, it is a peptide molecule produced by the probiotic organism, and it is having very good antimicrobial property. Bacteriocin and niacin, they have very good antimicrobial properties. So this bacteriocin and niacin can be used in the food production industry. So because of this presence of bacteriocin as well as niacin, we can inhibit the spoilage of the food. So that is the main importance of this bacteriocin. So the bacteriocin, they're responsible for destruction of the pathogenic cells. So this is the figure which shows how actually the bacteriocin is acting on the pathogenic cell and how it is damaging the cell wall of the bacterial cell, pathogenic bacterial cell, and how actually it is completely destructing the pathogenic organisms. This is the bacteriocin activity against different pathogenic organisms. The 3 plus shows the maximum activity. So we purified the bacteriocin, and the pure bacteriocin we used against the pathogenic organism, and the bacteriocin has shown very good effect against the pathogenic organism. we studied about their lactic acid, because lactic acid, so increase in the concentration of lactic acid decreases the pathogenic microorganism because of the acidic nature in the intestine, or because of the acidic nature in the foods whatever we are storing. So there is less possibilities of growth of microorganism. More the acidic condition, more the destruction effect against the pathogenic organisms will be there. So we have seen the concentration of lactic acid produced by this organism. So each organism they produce around 0.5 to 7 gram per liter of lactic acid. So that means the organism got a very good property to inhibit the pathogenic organisms in the intestine. Lastly, we've seen about their this one hydrogen peroxide. The hydrogen peroxide it is prepared by the organism as a chemical, as a toxic chemical. But this hydrogen peroxide, which is produced by the bacteria, this one by the probiotic organism, it is not affecting the probiotic organism, but it is affecting the other organisms. So generally, it can cause the cell damage effect, or it can act as a free radical so that it can destroy the this one the pathogenic organism. So we have seen the concentration of hydrogen peroxide produced by the organism. They will produce a little concentration of this one hydrogen peroxide, but even though the concentration is very less, around point, zero point 0.07 gram per liter, but this much hydrogen peroxide which is produced by this organism itself is enough to act as a antimicrobial agent. So overall, I told in the beginning, so finally we done the molecular characterization, and these are the 22 organism we identified. We isolated 55 organism, total 55 from 15 non-fermented food sources. Out of that 55, we have done the sequencing, 16S rRNA sequencing to identify the proper organism. So we have done 22 organism, and the sequences has been submitted to NCBI. And this is the frequency of the isolates in different sample. That means some sample they got more number of probiotic organisms, some sample, some non-fermented sample they got less number of probiotic organisms. So this is about their frequency of availability. And in conclusion, we can say that from 15 non-fermented food products, we have isolated 55 lactic acid bacteria, and almost all the this one probiotic organism. They exhibited very good properties of very good properties of resistance against acid, bile, GI tract environment, aggregation, co-aggregation, adhesion, etc. So this property shows that they are the potent probiotic organism which can be used in our regular diet to get a good health. So this was the conclusion. And in the recommendation, you can say that generally what people do, the probiotic or the lactic acid bacteria, means a general thought is that the probiotic or lactic acid bacteria they are present in the non-fermented this fermented food, maybe curd or maybe pickle or some other varieties of fermented food. But what we can do is, even though they are present largely in the fermented food, we can also find their presence in the non-fermented food. So very less research has been done to find out the presence of this probiotic organism in the non-fermented food. So this was our aim, to find out the presence of this probiotic organism from the non-fermented foods. So we could get around 55 good probiotic organism from this non-fermented food. And it is also important to explore the probiotics from unusual sources. So generally the probiotics means these are the environmental contaminants. They can be present anywhere on any surfaces. So we want to find out them from any unusual sources other than this fermented or non-fermented food. We can also find out from any unusual sources. And currently there are very few probiotic supplements are available in the market. So such type of probiotics which we are isolating and we are identifying, they can be purified, they can be finally commercialized. So that was the recommendation. And I'm thankful to ICMR. So this project was funded by ICMR, and we got a good grant for this project. And these are the references. And thank you. If any questions are there, welcome.
Thank you so much, sir. Anyone asked to any questions to our guest speaker? You can ask directly or raise your hand or text a message. No, sir.
Yes,
Thank
Mr.
you, sir.
Chetan.
Yes, sir.
Mr. Siddharth.
Yes, sir. हेलो।
हाँ जी सर।
ये सर बताइए सर, कुछ बता रहे थे आप।
जी ये कंप्लीट हो गए थे तो हमने कहा सर का कुछ नेक्स्ट प्रोसीडिंग क्या है सर?
सर नेक्स्ट प्रोसीडिंग स्पीकर्स हैं, इंटरनेशनल स्पीकर हैं, डॉक्टर उस्मान जैंगो सर। Okay, I want to express my sincere gratitude for the opportunity to stand before you today and share insight during the lecture by Dr. Nannu Safagatullah sir. Your presence and engagement have made this experience truly meaningful. Thank you for your thoughtful discussion. I appreciate your time and attention. Let's continue to learn and grow. Now, our next guest speaker, international guest speaker, Dr. Usman Umar Jango, sir. I hope, sir, you are joined and you listen.
Yes, sir, I'm here. Are you hearing me, sir?
Okay. Dr. Usman Umar Jango, sir, currently working as Associate Professor and Head with Department of Biological Science, Sadatu Rimi University of Education, Combusto, Kano State, Nigeria. Dr. Jango interest range for bioremediation of heavy metals, microbial drugs resistance to antibiotics, public health and water and environment sanitation through creation awareness in issue on related to such as National Agency Food and Drug Administrative and Control, Standard Organization of Nigeria as well as Kano State Consumer Protection Agency. He also participated in curriculum development and national commission for colleges of education. Dr. Jango published many research papers with high impact factor and attended several national and international conferences. Now I would like to warmly welcome Dr. Usman Umar Jango, sir. Please, sir.
Yes, sir. Are you hearing me?
Yes, sir. Very well.
Okay. First of all, I would like to thank the organizers of this international conference and then also the participants. I would like to present my topic for this conference, phytochemical screening and antimicrobial activity of Jatropha tangerensis against some clinical isolates. Then the abstracts, the leaves of Jatropha tangerensis were
So
collected.
actually, we cannot see the PPT. Is there any PPT?
Yeah, I have sent the CPPT for you. Are you hearing me?
Yes, sir. Okay. I will share it. I will say it, sir.
Okay. The leaves were of the Jatropha tangerensis were collected and assayed for phytochemical composition and the microbial activity using methyloric and hexane extracts as determine the bioactive component of the effect of the organism. Phytochemical screening was carried out and determined the proximate composition using standard procedure, which revealed the presence of sapronine, glycoside, reducing sugar, steroid, alkaloid, and flavonoid. Then the extracts were used using methodic and water solvent. Then antimicrobial activity of the leaves were carried out, and they show that the microorganism, which is a Salmonella and Escherichia coli, were sensitive to the extract. Then introduction. The Tupora gentiraceae belong to the family Eoporiaceae, which is a common shrubs, 0.8-meter height, and it is usually grown in the forests of the West Africa and used as a source of food, ornamental, and medicinal purpose. Many scientific evidence reveal that the medicinal properties of this Gentrophora tendronensis, and this the plant has been reported to carry some phytochemical constituents that lower the blood cholesterol level, and also it is used for the treatment of cardiovascular diseases, and also it is used for hyperlipidemia, as reported by Artesania et al. 2012. It also received a lot of attention in potential of health due to its availability and affordability. The phytochemical screening of Jatropora tuberosa leaves revealed it contained bioactive principles such as alkaloid, flavonoid, tannins, glycoside, and anthraquinone and sapronins. The pharmacological studies reveal that the plant shows some wide range of biological activities, which include hypertensive, antioxidant, antimicrobial, antimalaria, hypoglycemic, hypolipidemic, and hematological activity. Although there were growing popularity of herbal medicine as a safe, but scientists still advocate for proper physiological and dosological tests in order to ensure safety and use of traditional medicine. In view of the comment, the Jatropora tendronensis as a traditional herbs therefore need proper physiological dosological tests in order to ensure safety and dose, as reported by Oguroye and Korime 2022. The aim and objectives of the study is to determine the physiological screening and antimicrobial activity of Jatropora tendronensis against some clinical isolate. So in this view, the following objectives were drawn. Number one, to screen out the phytochemical content of Jatropora tendronensis leaves extract. Number two, to determine the antimicrobial activity of the leaf extract, Jatropora tendronensis against some clinical isolate. And then to determine the minimal and inhibitory concentration of MIC and minimum bacterial concentration. Now, materials and methods. The study were carried out in Albasu local government area of Kano State, Nigeria. The fresh sample of the Jatropora tendronensis were collected from a healthy, fully grown plant from Albasu local government, Kano State. The leaves were identified by the expert in the Department of Plant Science, Bayero University, Kano, with herbarium access number BUCAN 0584. The leaves were washed with distilled water and kept under dry conditions in the laboratory. 100 grams of the weighing powder dry leaves sample were partitioned using solvent, ethanol, and water at 70 to 80 volume by volume. The sample were poured in a 1 liter separating funnel covered and shaken every day for 1 week. The solution were subsequently tracked and filtered using Wattman filter paper number 1 and filtered. then evaporated to dry using rotary evaporator. The extraction were stored at 4 degrees centigrade as reported by Akinyemi and at ambient temperature as reported by Akinyemi and Sharma 2018. The extra were dissolved in 30% diameter sulfide DMSO, and further dilution were carried out, were obtained of 100 milligram per liter, 50 milligram per liter, 25 and 12.5 milligram per liter concentration and stored at 15 degrees until required for use. Then phytochemical screening were carried out with the following alkaloid, sapronine, tannins, steroid, flavonoid, and resins as reported as using standard procedure. I don't have to explain all the procedures. Then 2 clinical isolates were obtained from the pathological department of the General Hospital, Wudil, Kano State. The isolate were taken to the laboratory of microbiology department, Aliko Dangote University of Science and Technology, Wudil, Kano. The isolate were confirmed using gram staining and other biochemical tests in order to actually identify the microorganism, the isolate we are going to use in the work. Then determination of minimum inhibitory concentration were carried out. Extract were evaluated by the method as described by Yagana et al. 2012. Extract of the concentration 5 milligram per liter were serially diluted with the distilled water to various concentration of 12.5, 25, and 50 and 100 milligram per. The extract were mixed in using were evaporated using equal test in the using nutrient broth, and equal proportion of the sterile tubes were proportion of 0.5 milliliter of the culture media as added, and the tube incubated at 37 degrees centigrade for 24 hours. The latest concentration were shown in the visibility gross bacteria as taken as MIC. The determining of minimum bacterial concentration were also carried out using stand as reported by Ismon et al. 2017. This is the result. The result showing that in the ethanol extract, there is a presence of tannin, flavonoid, sapnoid, terpenoid, and carbohydrate, while in the presence of aqueous extract, there were no presence of terpenoid and carbohydrate. Yeah. And then in the table 2, which shows the zone of inhibition of the thoroughly extract against Salmonella typhi and Escherichia coli, and it shows that the highest zone of inhibition were obtained at 100 milligram per liter, which is 27 against Salmonella, while the highest zone of inhibition against Escherichia coli were 20, and then a control of using antibiotic supply were obtained of zone of inhibition of 22.19 zone of millimeter against Escherichia coli, while against Escherichia coli, while against is 22.19. Then the zone of inhibition of aqueous extract against Salmonella typhi and Escherichia coli were obtained, where the highest zone of inhibition against Salmonella typhi were obtained about 22 millimeter, while the zone of inhibition, highest zone of inhibition, were obtained using aqueous solution at 2.14 millimeter. 14 millimeter. Then the minimum inhibitory concentration against aqueous and show that there is a growth of the bacteria at 25 and 12.5 milligram per liter, and then 25 milligram per milliliter. And also the minimum inhibitory against ethanoid is 12.5 milligram per milliliter. The discussion the Jatropha temperature which contain flavonoid, saponoid, and carbohydrate as the major component of as shown in the table 1. The results also reveal that the medicinal leaf, the leaves of Jatropha tendrensis contain certain metabolites such as tannins, albanoid, and flavonoid, which are rich in interfering with the protein synthesis in the bacteria, thereby inhibit the protein synthesis of the microbes, as reported by Sachs et al. 2015, Suchira 2016, and Cowan 2019. Flavonoids are known as the produce plant that respond to microbial infections, which affect intensive studies were found to have antimicrobial activity as reported by Cowan 2019. Saponins, which are glycosides have been found to inhibit effect. Also, flavonoid, in other hand, are phenols in nature, which due to they affect the nurture and coagulate the microbial cells, and as a result of that, it will inhibit the growth of the microorganism. Then it was also found that the antimicrobial activity of ethanoid extract of Jatropha tendrensis against Escherichia coli and Salmonella. This is similar with the Vera et al. 2015, which report Tendrera extract have effect against inhibiting Escherichia coli and Salmonella and Staphylococcus aureus. In et al. 22 that significant inhibitory activity against the growth of the microorganisms, Escherichia and Salmonella type against ethanoic extract of Jatropha tendrensis. The antimicrobial activity may be due to the phenolic compound, which is responsible for antimicrobial activity of the plant, as reported by Kisbro 2014. Both two extracts of the Jatropha tendrensis have inhibitory effect against the bacterial species of E. coli and Salmonella, but the ethanolic extract is more effective in antimicrobial activity compared with the aqueous extract due to ability of the solvent to diffuse easily into the cell and its component, as reported by Mahfouz Haq et al. 2002. In conclusion, the study it reveals that the Jatropha tendrensis contain tannin, flavonoid, saponoid, and terpenoid. Also, the two ethanolic and water extract of the plant were found to be effective against Escherichia coli and Salmonella. The findings of the study recommend that the dose extract to be used should be 100 milligram per liter concentration. More antibacterial screening tests should be carried out against different white bacteria in order to find out if it is against positive and gram-negative bacteria. Then further research should be carried out on the antifungal property and toxicity test of the extract. Thank you very much. Thank you very much. Hello.
Thank you very much, sir, for outstanding deliberation and application of the herbal phytochemicals and bioactive compounds for scientific aptitude and eradication of certain target diseases caused by bacteria. So it was an outstanding presentation, and we are highly appreciative for sparing time with us from your busy schedule from an international pole from further India. So we are highly grateful to you, sir, and thank you so much, sir.
I'm very, very grateful, sir. Thank you very much. Thank you very much. I really feel honored for giving me the opportunity to present my research. Thank you very much.
Thank you so much, sir. It's a very nice and fruitful lecture. Now our next session will start 11:45 and two international speakers are here from 11:45 to 1 o'clock. And this half an hour we will arrange oral and poster presentation. The name is Avilas Sahu and Sarika Bhusan from BVAU Lucknow. Avilas Sahu is he Avilas Sahu?
Yes, sir.
Yes, sir. Please present your paper.
Okay, sir. Is it visible?
Yes, sir. Yes, sir. Visible, visible.
Very good morning to everyone present here. My name is Abhilash Sahu, and I am currently an MSc Zoology student from BBAU, Babasaheb Bhimrao Ambedkar University, Lucknow. So my topic of presentation is Advancement in DNA Repair Technology, Unveiling the Future of Genetic Health. So coming to the introduction to DNA repair, DNA damage is a detrimental effect on the primary structure of the DNA. It can be categorized into two types. It is endogenous and exogenous type. In endogenous, it occurs when DNA reacts with water and other ROS species like reactive oxygen species, which leads to many hereditary diseases and cancers. In exogenous damage, it occurs by environmental, physical, and chemical agents like ionizing radiation, UV radiation, alkylating agents, and cross-linking agents. So the process of rectifying this damage is what we call as DNA repair. So DNA repair happens in our own cell by various methods like BER, which is base excision repair. It corrects small scale damages like base modification or single base damages, which may be caused by oxidation or alkylation. Then we have NER, that is nucleotide excision repair, which fixes bulky lesions, including UV induced damage and chemical adducts by removing a segment of damaged DNA and replacing it with the correct DNA sequence. Then we also have MMR, which is mismatch repair. It corrects errors that occurs during DNA replication, and it ensures that base mismatches and insertion or deletion loops are fixed. So the DNA integrity is very important for our genetic health. It is helpful in preventing mutation, proper cellular function, and also in proper inheritance of characters, maintaining homeostasis in the cell. So this is responsible. The DNA. So that is why DNA integrity is very crucial. Then coming to the challenges which are associated with DNA damages are the aging, cancer, impaired reproduction. Sometimes what happens is the DNA of germ cells get damaged, which may result in infertility and genetic defects, which may further go on passing to the offspring, which impacts the future generations. So other mutations and genetic diseases can also be a result of DNA and damage. Coming to the CRISPR-Cas9 precision genome editing, it is a gene editing technology which helps in precise modification of DNA. Here, CRISPR stands for clustered, regularly interspaced, short palindromic repeats. These are the segments of DNA containing short and repetitive base sequences which are derived from various microbes, especially the viruses and other invaders that has previously attacked the organism. So the CRISPR sequences are a part of bacterial immune system and they act as a memory of our past infections. The Cas9 is a kind of enzyme. It acts as a pair of molecular scissor. It is guided by a specific RNA molecules called gRNA. So gRNA is also called guide RNA. These guide the Cas9 to specific locations on the DNA that match the sequence which the gRNA has. So once the Cas9 reaches there, it can cut the DNA at that precise location. So coming to its working, scientists design the gRNA sequence complementary to the target DNA, which they want to modify. When this gRNA is introduced along with the Cas9 enzyme into the cell, it guides the Cas9 to the exact spot of the DNA. Then Cas9 makes the cut at the location, removing the defective DNA sequence. So it has many applications, including in especially in gene editing, in biotechnology, and many other applications are there of this Cas9 system. Now coming to the DNA nanotechnology and origami. So DNA nanotechnology and origami techniques are innovative methods. These are used to design and manipulate DNA molecules into precise shapes and structures. So and the concept involves engineering DNA molecules to form specific nanostructures, sometimes resembling the origami shapes. As we know, origami is an art of is a Japanese art of folding paper into various structures. So similarly, the DNA is folded into various nanostructures to serve various purposes, like which include drug delivery, sensing, and aiding in DNA repair. So and this was about the DNA nanotechnology and origami. Now coming to the artificial intelligence in DNA repair, AI plays a very significant role in predicting, identifying, and potentially repairing DNA damage. Coming to the prediction part, they analyze vast amount of genetic data to identify patterns which are associated with specific DNA damages or mutations. Then coming to the identification of DNA damage, the AI assist in detecting and identifying different types of DNA damage from various sources such as radiation, chemicals, and oxidative stress. So they analyze the images and pattern using algorithms which help researchers and clinicians to accurately identify the DNA lesions or abnormalities in the genetic data. They have various applications in drug discovery and development, in precision medicine, and all these kind. So coming to the examples of AI powered tools which enhance our genetic health, the tools like Watson and VS Clinical use AI algorithms that interpret genetic variants. Platforms such as DNA Nexus and Seven Bridges utilize artificial intelligence to analyze a large scale, huge amount of genomic data, which helps the researcher to manage, analyze, and interpret genetic sequences. Then AI-driven drug discovery platforms such as Atomwise and Benevolent AI use machine learning to predict molecular interactions. They also help in designing novel drugs. And there are also companies like 23andMe and Color Genomics, which use AI to analyze genetic data, and they also assess the individual's risk for various genetic conditions and diseases. Then there are also tools like Foundation One and Tempus, which use artificial intelligence to analyze tumor profiles, and they can also make a personalized cancer treatment for the patient. Then there are also AI-driven platforms like Synthigo and Inscripta, which helps researcher to optimize CRISPR-based gene editing. phase 2 gene is a special kind of AI power diagnostic tool which analyzes the facial features of the patient to aid in diagnosing rare genetic disorders which the patient may inherit. So this was about the AI powered tools and platforms that are revolutionizing the genetic health. Now coming to the conclusion, so the advanced DNA repair technology, they have a vast potential for revolutionizing and the genetic health in many ways, like prevention of diseases and treatment, then precision medicine, and aging and longevity. DNA damage accumulation is linked with aging process. So advanced repair technology could potentially slow down that or even reverse the aging related effects by maintaining genomic integrity. So this also helps in cancer therapy and enhancement, then gene editing and gene therapy. So this was about my topic. So thank you.
Thank you, Avilas. Next presentation, Sarika Bhusan. Sarika Bhusan.
Good morning, sir.
Yes, please present your paper.
Yes. Sir, my screen is visible.
Yes, visible.
Yes, sir. Good morning, sir. Morning, everyone. Myself, Sarika Bhushan, studying in MSc Zoology, semester third from Babasaheb Bhimrao Ambedkar University. Here to present an oral presentation on the topic metabolic profiling of medicinal plants traditionally used against microbial infection. Here are the contents which I'm going to discuss about. So, firstly, we will go through the medicinal plants. What are medicinal plants? Medicinal plants are those which contain substances that can be used for therapeutic purposes or for the synthesis of useful drugs and medicine. Now we'll go through now benefits and uses of the medicinal plants. Benefits and uses are hereby we can use as treatment for common ailments in which medicinal plants like ginger and peppermint are used to address common health issues such as cold, coughs, headaches, wound healing. Aloe vera and calendula with anti-inflammatory, antimicrobial properties traditionally promote wound healing for minor cuts, burns, and abscesses. Pain relief. Willow bark, historically known for its analgesic properties containing salicin. Next is anti-inflammatory agents. Turmeric and ginger, recognized for their anti-inflammatory properties, find applications in managing conditions like arthritis. Antimicrobial and antibacterial agents. Garlic and tea tree oil, plants producing compounds with antimicrobial properties. Regulation of blood sugar. Medicinal plants like bitter manal, digestive health. Peppermint and fennel are used to soothe digestive discomfort and promote gastrointestinal health. Stress and anxiety reduction is also useful. Cardiovascular health, respiratory health. These are in which your medicinal plants are used to heal all these problems. Now we'll come to the metabolic compound. Following are the medicinal compound: alkaloids, terpenoids, flavonoids, phenolic acids, saponins, tannins, lignins. Alkaloids, these are the nitrogen containing compounds with diverse pharmacological activities. Terpenoids are the large and diverse class of compounds derived from isoprene units. They include essential oils and have various pharmacological properties. Flavonoids, these are the polyphenolic compounds with antioxidant properties. Flavonoids are found in wide variety of fruits and vegetables and medicinal plants. Phenolic acid, these compounds have antioxidant properties and can contribute to overall health benefits of saponins. These are the glycosides with soap-like properties. Saponins have diverse biological activities, including antimicrobial and anti-inflammatory effects. Tannins, these polyphenolic compounds have astringent properties and are found in plant tissues. Tannins have antioxidant and antimicrobial effects. Example, tea tree oil containing tannins compound. Numerous of these metabolites functions as bioactive substances and can be hostile to specific bodily functions. Now we'll come through the structures of these compounds. These are the alkaloids, these are terpenoids, flavonoids, phenolic acids, saponins, tannins, and lignans. Uh, these metabolic compounds can be extracted through these methods. Following are the methods: macerations, infusion, decoction, steam distillation. First we'll go through macerations. In this is the simple and traditional method where plant material is soaked in a solvent at room temperature for an extended period. The solvent extracts the bioactive compound from the plant material, resulting liquid, and the extract is filtered. Infusion. Similarly to macerations, infusion involves steeping plant material in hot water or solvent for a shorter period. This method is commonly used for extracting compounds such as flavonoids and phenolics. Then we'll go through steam distillation. Steam distillation is employed to extract volatile compounds, particularly essential oils. Steam is passed through the plant material, causing the volatile compound to evaporate. The steam is then condenses, and the resulting mixture of essential oil and water is collected. Solvent extraction. This method involves using organic solvents, example ethanol, methanol, to dissolve and extract the bioactive compounds. The solvent is then evaporated, leaving behind the concentrated extract. Multiple rounds of extraction may be performed to enhance yield. Next, a few methods also used in the extraction method, like Zoxalate extraction, supercritical fluid extraction, ultrasound assisted extraction, microwave assisted extraction, solid phase micro extraction. Then we'll go through the analytical methods, which includes chromatography, mass spectrometry, nuclear magnetic resonance, capillary electrophoresis, liquid chromatography, mass spectrometry imaging, Foreign transform infrared spectroscopy, these options electrospray, ionization, mass spectrometry, high resolution mass spectrometry. Metabolomics platforms, which includes GC-TDF-MS and LC-QT-DF-MS. First, we'll go through the GC-TOF-MS gas, which is gas chromatography, time of flight mass spectrometry, and LC-QTOF-MS liquid chromatography, time of flight mass spectrometry. Then last one is targeted and untargeted approaches. Targeted metabolics include quantifies specific known metabolites, and untargeted includes profiling as many metabolites as possible without prior knowledge. The choice of methods of these analytical methods depends on the research objective, metabolite types, and the material plant material characteristics. Now we'll go through the uses in modern medicine of these medicinal plants like tulsi. Tulsi is holy basil, traditionally used in Ayurveda. Scientific interest in modern medicine. Antimicrobial properties having against bacteria, viruses, fungi. Anti-inflammatory effects also, cardioprotective effects, respiratory health benefits incorporated into pharmaceutical and nutraceutical formulations. Next is betel leaf, which is also commonly known as pan. Traditionally medicinal use, recently scientific exploration is also going. Anti-inflammatory and analgesic effects from polyphenol and flavonoids, historical use of oral health, respiratory health, investigated for antimicrobial properties against bacteria, fungi, and viruses. Caution required due to association health risk, including oral cancer. Next is marigold leaf. It is used traditionally as medicinal properties, wound healing properties with anti-inflammatory and antimicrobial effects, anti-inflammatory effects for conditions like dermatitis, antioxidant properties from flavonoids and carotenoids, potential anti-cancer properties, and anti-arthritic effects used in ointments, cream, and extract for various medicinal purposes. Ginger, this anti-nausea property for pregnancy, chemotherapy, and surgery. Gastrointestinal health benefits is also included in this. Respiratory health, possible role in weight management also. Demonstrated antimicrobial properties, ongoing research is going on this. And then we will go through papaya leaf. Papaya leaf, it is traditionally used uh, in the treatment of dengue fever, which increases platelets production. Immune system support via immunomodulatory properties, antioxidant properties for neutralizing free radicals, investigated for potential anti-cancer properties, ongoing research for efficacy and safety in various health conditions. Oregano leaf, uh, it has also medicinal properties, strong antimicrobial properties from essential oils, respiratory health supporting conditions like infections, gastrointestinal health benefits, anti-inflammatory effects, antifungal properties, and these also clinical research
Please
is
wind
going
up.
on. Yes, sir. These are the leaves, plants of these, and here are the limitations of this metabolic profiling, and this is the conclusion. Hereby, the metabolic profiling of medicinal plants employs advanced analytical techniques like chromatography and etc. And these are my references. Thank you, sir.
Thank you. Next one is Abhishek. Abhishek De. Every participant, please confirm that your presentation time is 5 to 10 minutes, not more than. Next one is Abhishek De. Please bind up your presentation
Good
within
morning,
5
sir.
minutes, 5 to 7 minutes.
Good morning, sir.
And our next guest speaker is Dr. Navjeet Kaur, ma'am. So our next guest is, lecture will be start within 5 to 7 minutes. Please bind up within 5 to 7 minutes, Abhishek.
Yes, sir. Sir, is my screen visible?
Yeah, it's visible.
Okay, sir.
Uh,
Okay, good morning, everyone. I'm Abhishek Dev from Department of Zoology, BBU, Lucknow. And today I'm going to present a topic topic on the use of AI in analyzing cell division and in control of cell cycle. So let's know a little bit before starting this. Let's know a little bit about AI. So AI took birth during 1952 to 1956, and it has improved ever since till now. At this present time, it was actually named by John McCarthy in 1956 at the Dartmouth workshop. So at the modern time, AI is very much advanced, and it is almost used everywhere. Our that personalizations that we see in our phone, and the face recognition feature, the self-driving cars, these are all actually self-driving cars. These are all advanced approach and advanced stage of AI. So AI, this indulgement of AI can be also done in analyzing cell cycle and cell division. So, talking about cell division, cell cycle is a process from which cell prepare to divide, and it actually divides, and it happens in a cyclic manner. There are daughter cells are formed, and these daughter cells again undergo the same preparation and division process. Various cell stages are there in the cell cycle, such as G1, S, G2, and M. M phase again has prophase, metaphase, anaphase, and telophase, so on. So some features of AI are there, such as machine learning, deep learning, etc., by which we can identify the various stages of cell cycle and cell division process. We can basically train the AI tool, and after training, when it becomes efficient, we can prepare a model, and again we can test that model to whatever we have trained for. For example, we have trained for detecting a specific type of cell. Then after training, we can start testing on the AI tool on detecting that specific type of cell in a heterogeneous cell population. For example, we have a sarcoma cell. If we have trained our AI tool to detect, then if in a sample there is sarcoma cells present, our tool should be able to detect it, and it actually happens. And use of there is a research paper published by Yukiko Nagao and others. They have used CNN-based classifiers to extract features from the fluorescence microscopic images. It is a deep learning process, CNN is a deep learning process, and it is an image-based process. Here we used images to train the AI tool so that it can detect. Here we are using actually certain things like Hoch's DNA Golgi apparatus, that is GM130, and the microtubule cytotoxicity EB1. So in this, they demonstrated are classified in G1, S, G2, and other cell cycle phases. The AI tool was able to actually classify with these images that this particular cell is in this stage. So classification were based on various features of the different phases of cell cycle. Like in S phases, there is doubling of the content of DNA, and there is doubling of Golgi apparatus also. And during G2 phase, there is more mobility of microtubules so that it can go to the M phase. So the AI tool was able to detect all those and decide likewise. For training, cells were stained with the Hochst and an antibody to the cis Golgi matrix protein GM130 and the microtubule plus N binding protein EB1, as well as antibody to the G2 marker proteins CENPF. After training, the CNN model were able to distinguish the G1S and G2 phase only based on only Hochst staining alone. So it was able to distinguish between different cell cycle phases, not only on HeLa cells but also on other types of cells, and that accuracy rate was 90%. So, but other than Hochst staining, when it came to spatial patterns in the Golgi apparatus and the microtubule percent represented by GM130 and EB1 respectively, it gave around 80% of accuracy. So we can see in this research that Hochst training was much more, but it gave more accurate information. And now coming to the tracking cell division using AI, tool named cytosensors was developed by Martin Hellstone and few others, which is based on AI technology. It detects the stem cells and track their division in 3D, revealing how tissue changes over time. So scientists actually observed larvae of fruit flies. They actually observed the brain cells here. The tool cytosensors, it worked on the basis of machine learning, which is also, so in machine learning it was supervised type, so lots of training and testing were involved here. It also cytosensors was also applied on zebrafish organoids, and it was so that this tool that they developed was also not only compatible with one types of cells but also in many types of cells. Similar, another tool was developed by QSpert, that is DetectV, but it used a deep learning platform, and it did a cell division tracking in an automated way. So AI tool dynamic for measurement of single cell volume. We have talking about cell cycle checkpoints. There are many various checkpoints during a cell cycle, which also plays a major role that cells are ready to divide or not. So it is, if we get to know that okay, there is a malfunction in this particular checkpoint, then we will be able to diagnose many things. So here in this particular slide, we will talk about that this CTRL is able to detect the single cell volume. Okay, the size of the size and volume of the cell. So this AI tool actually used the differential interference contrast microscopic images, and when once trained, this tool was able to track single cell volume dynamics, and it was able to give a quantitative accuracy. This method helped for continuous single cell volume measurements in multiple cell culture forms without time limit. Actually, we can use this tool for arbitrarily for a longer time. And since we know that any malfunction in cell cycle can lead to cancer, so this AI tool can help, and knowing AI is, knowing cell cycle, detecting and understanding cell cycle via AI can prove more beneficial for us to get a proper treatment plan for cancer or maybe diagnosis also. So NCI researchers have made and tested AI algorithm that can identify cervical precancerous cell that can be tested and removed. We can basically include any diagnostic machine such as MRI, CT scan, this AI tool we can include so that we can get a better result. Actually, what happens is actually this AI tool helps radiologist. There are some non-cancerous cells which actually looks like cancerous cells, and if that radiologist is not particularly trained very much better, then it will be confused and might give a false result to us. But this AI tool is very efficient, and it can actually help radiologists to differentiate between cancerous and non-cancerous cells that look like cancer. So it can also help us to come up with a better treatment plan, as Dr. Hamad and her colleague built a deep learning model that was able to tell that this particular bladder cancer patient might need some other thing other than surgery, such as chemotherapy. What happens that in this bladder cancer, 50% of the cases, it happens that there are very small precancerous cells are there, or those cells which cannot be detected very easily, but AI tool is able to detect that. If AI tool can detect, we can apply this AI tool on that patient, and that AI tool is able to detect that. Then it can be also decided, okay, this particular patient need something more other than surgery, let us chemotherapy and all. So I would like to conclude my topic by saying that AI tool is very efficient in analyzing cell cycle, and it is very fast and less time concluding. It does not get tired very easily. It requires very less labor, as in deep learning method we have seen. AI tool can give quantitative accuracy and requires minimal sample preparation. We don't need much chemical, and very less samples are required. It applies on a wide range of biological and experimental conditions. AI can be a better tool for drug designing, and can also be more precise in diagnosis, and with more advancement, it can design more cost-effective treatment. So thank you very much. These are my references. Thank you so much.
Thank you so much, Abhishek. Now our guest lecturer already have joined in this session. So now I would like to invite Dr. Navjeet Kaur ma'am. Dr. Navjeet Kaur ma'am.
Am I audible? Good afternoon.
Yes, ma'am, you are audible. I am just introduced Dr. Navjeet Kaur ma'am. Dr. Navjeet Kaur ma'am received the BSc degree from Punjab University, Chandigarh, MSc and PhD degree in chemistry from Banastale Vidyapit under the supervision of Professor D. Kishore. She worked as an assistant professor in the Department of Chemistry, Banastale Vidyapit, and has started a specialization research career focusing on organic synthesis and medicinal chemistry. At present, she is working as an associate professor in Department of Chemistry and Division of Research and Development at Lovely Professional University, Punjab. Dr. Kaur has guided 4 PhD students and many MSc dissertation students. With 12 years of teaching experience, she has published more than 185 scientific research papers, review articles, book chapters, and monographs in the field of organic synthesis in national and international journals. She has published 13 books under the umbrella of reputed journals CRC Press, Taylor and Francis Group, Elsevier, Springer Nature, etc. She has featured consistently for 6 years in the world ranking of top 2% scientists in subject-wise analysis conducted by a team of scientists at Stanford University, United States. 2020 to 2023 full career and single year ranking, 2019 full career ranking, 2018 single year ranking. Numerous newspaper has reported on her achievement of top rank in such a short span 5 to 6 year of her career. She is the recipient of many more awards. In 2021, Dr. Kaur was selected as a candidate for membership by Royal Society of Science Research, London, England, UK. She has been certified for her contribution to science and technology on Women's Day by DST Government of Rajasthan. She is currently serving as an editor-in-chief for Advanced Chemicobiology Research and editorial board member of various reputed journals. She has attended about 50 conferences, workshops, seminars, and refresher courses and delivered many invited lectures and radio talks. She acted as a reviewer for many papers in different journals. She has served as a program co-chair and member of organizing committee of conference at Akal University, Punjab, in collaboration with the Indian Chemical Society and Royal Society of Chemistry, London. In addition, she was worked as National Serve Scheme Program Officer from 2016 to 2022 and a member of Unnat Bharat Abhiyan from 2018 to 2022, involving in the basis daily needs, healthcare, education, environment, sustainability, and agriculture activity in rural areas. Dr. Navnit find interest in Sikh literature and has completed a 2-year Sikh missionary course from Sikh Missionary College, Ludhiana, Punjab. Now I would like to warmly welcome Dr. Navjeet Kaur ma'am. Please, ma'am.
Yeah, thank you so much for the introduction. Okay, my screen is visible to you.
Yes, ma'am. Very much visible.
Okay. Today I'll be talking about his synthesis of chemotherapeutically useful agents, right from 1,3-dihydro-2H1,4-benzodiazepine, class of privileged medicine scaffolds. Firstly, the overview will be, firstly we will talk about the introduction, then the preparation of starting materials, then synthesis of oxidizole, imidazole, benzimidazole, thiazole, isoxazole, and pyrazole incorporated analogs of 1,4-benzodiazepines linked to it through 2-phenoxyl or phenylamine bridge. And then it will be synthesis of primido incorporated analogs of 1,4-benzodiazepines linked to it directly or through a 2-phenoxyl or phenylamine bridge. As a last section will be synthesis of benzodiazepine, benzoxazepine, incorporated analogs of 1,4-benzodiazepine linked to it through 2-phenoxyl bridge. Introduction. Hepatocycle chemistry actually is considered as the largest division of organic chemistry, and more than half of the chemical entities which are known, they are comprised by heterocyclic compounds. Majority of the pharmaceuticals, whether they are natural or synthetic, there is a pesticide, insecticide, vitamins, and dyestuffs. They contain heterocyclic rings. And many of the compounds synthesized in the industrial sector each year, they are heterocyclic in nature. The heterocyclic compounds have different uses. They can be used as a structural motif for new materials, can be used as a protecting groups, as unique solvents, as a reagent. Even they can be used as a functionalities in organic synthesis. They are the chemical entities which can be modified or substituted. If we talk about the synthesis of heterocycles, many methods for the synthesis of heterocycles are classical in nature. We can say they are traditional, and mostly the synthesis involves some type of condensation reaction. Modern examples also exist, and they provide different substitution patterns on the ring. Ideal synthesis for heterocyclic compound will be the one in which the product will be obtained in few steps as possible, in a good yield, and by using environmentally benign reagents. The synthetic variables that need to be optimized, they are the time, cost, overall yield, simplicity of the performance, safety, and the environmental acceptability. Our main nucleus will be was the 1,4-benzodiazepine nucleus, and 1,4-benzodiazepine nucleus is known to exhibit a wide range of pharmacological properties. And along with 1,4-benzodiazepine, there are some condensed heterocyclic rings such as oxidizole, imidazole, benzimidazole, thiazole, isoxazole, piozoles, primidines, 1,5-benzodiazepine, 1,5-benzothiazepine, and 1,5-benzoxazepine nuclei, which have also attracted the attention of chemists on account of being medicinally active. And in our work, we had combined both 1,4-benzodiazepine nuclei and the other nuclei which I have just discussed. Now, if we talk particularly about 1,4-benzodiazepine nuclei, 1,4-benzodiazepine can exhibit anti-tumor, anti-cancer, anticonvulsant, antidepressant, anti-HIV, as well as anti-anxiety properties. These are just a few to mention. And the objective of our this research was we have divided it into two portions. And the first strategy in the present work was to synthesize the 1,4-benzodiazepine nuclei and the nuclei in which there will be structure features of bioactive pharmacophores and those pharmacophores which have the previous history of being biologically active, with the expectation their presence in a single molecular framework contributes significantly to the biological activity in the formed molecules. As I mentioned that our main nucleus was 1,4-benzodiazepine, and on the 1,4-benzodiazepine nuclei we had appended different nuclei such as oxidizole, imidazole, benzimidazole, thiazole, isoxazole, piozoles, primidine, 1,5-benzodiazepine, benzothiazepine, and 1,5-benzoxazepine. In this slide, you can see there is the structure of tetravalent drug. Tetravalent is an anti-HIV drug, which is FDA-approved drug. And tetravalent drug has main three vital fragments. One is in red one. Red one is aminoprimidine. Then in blue is we have paracyanoxyphenide, and then pink one is a paracyanoaminophenoid. So these are the three main fragments of tetravalent. And our second strategy in our work was to incorporate these three substituents on 1,4-benzodiazepine nuclei. So our main nucleus is 1,4-benzodiazepine. On the second position of this 1,4-benzodiazepine nuclei, we had firstly we had appended phenoxyl and phenylamine group, which will act as a spacer, and then we through this spacer we had appended different nuclei. In among these there were 5-membered rings like this one oxidizole, imidazole, benzimidazole, thiazole, isoxazole, and pyrazole. And in next section, we had incorporated 6-member rings, which are mainly pyrimidine ring. And in the last section, we had incorporated 7-member rings, like 1,5-benzodiazepine, 1,5-thiazepine, and 1,5-oxazepine. The first section will be preparation of starting materials. These are the compounds whose synthesis had been carried out. Um, as you can see, these are the amidine compound with phenylamine or phenoxyl spacer. And these two are the immediate esters. And then is a thiocyanidone intermediate. Is a dimethyl luminoketone intermediate which we had synthesized. And finally we had synthesized the charcoals. And all these intermediates will be used for the synthesis of final compounds. If we talk about the schematic plan, we had started with acetone. And acetone molecule was reacted with chloroacetyl chloride to form N-chloroacetyl chloride, which was further reacted with hexamine in presence of methanol to synthesize our main nucleus, that is 1,4-benzodiazepine. And then 1,4-benzodiazepine nuclei is reacted with N-methyl are present. And after this, the ketone was converted into Cl because chloro is a good leaving group on reaction with PuCl3 in presence of a dimethyl aniline base. Then the synthesized 1,4-benzodiazepine nuclei with chloro substituent was reacted with different paracyano compounds to form a paracyano intermediate, which upon reaction with hydroxylamine hydrochloride given us the intermediate, that is imidazole, and in alternative reaction or on reaction with methyl alcohol and given immediate ester. If we talk we see this next branch, this chloro was reacted with para-hydroxy benzaldehyde, which gives us a benzaldehyde intermediate, which was further reacted with thiocyanocarbazide to give a thiocyanocarbazole intermediate. In the third branch, we can see that the chloro substituted 1,4-benzodiazepine was reacted with para-hydroxyacetophenone, and which gave us acetophenone intermediate, and which upon further reaction on the one hand with DMF gave us dimethyl intermediate, and on reaction with the benzaldehyde it gave us a charcoal intermediate. The next section was the synthesis of oxidizole, imidazole, benzimidazole, thiazole, isoxazole, and pyrazole incorporated analogs of 1,4-benzodiazepines linked to it through 2-phenoxyl or phenylamine bridge. Our aim in this section was to incorporate 5-member bioactive pharmacophores on the second position of 1,4-benzodiazepine molecule to see or to synthesize the new molecules which will have a pharmacological significance. These were the rings which were incorporated on 1,4-benzodiazepine nuclei: oxidizole, imidazole, benzimidazole, thiazole, isoxazole, and pyrazole. All these are 5-member rings. And we have synthesized these compounds. You can see is oxidizole incorporated analogs through phenylamino and phenoxyl group, and we have imidazole analogs of 1,4-benzodiazepine through phenylamino and phenoxyl spacer, and we have benzimidazole analogs. We had synthesized isoxazole analogs, and we had synthesized pyrazole analogs, and finally we had synthesized thiazole analogs of 1,4-benzodiazepines. To start with the schematic plan, we had taken the amidine intermediate which was synthesized under the section of preparation of starting material is reacted with acetyl chloride. And the difference between these two is that in this we have a phenylamine spacer and here we have phenoxyl spacer. And on reaction with acetyl chloride, the both give the oxadiazole analogs of 1,4-benzodiazepines. In this reaction and in the following reaction, this one, you can see that we have taken immediate ester. In both the reactions, we have immediate ester, which was reacted with ethylene diamine, and upon reaction with ethylene diamine, it gave us imidazole analog, but this imidazole was a reduced one. And further, it was oxidized upon reaction with iodoxybenzoic acid to give the imidazole analogs of 1,4-benzodiazepines through phenylamine and phenoxyl spacer. Over here, we can see that the immediate ester in both the reactions, immediate ester through phenylamine and phenoxyl spacer were reacted with orthophenyldiamine to give the benzoimidazole incorporated analogs of 1,4-benzodiazepine. When thiocyanocarbazole intermediate was reacted with ammonium iron sulfate, it gave us a thiazole analog of 1,4-benzodiazepine. Over here we can see that in both the reactions we had synthesized isoxazole analogs of 1,4-benzodiazepine. In first reaction, the intermediate taken was dimethylamine ketone, which upon reaction with hydroxylamine hydrochloride in presence of a base gave us the final compound. But in second reaction, the intermediate is a charcoal, which upon reaction with hydroxylamine hydrochloride gave us the isoxazole, but this isoxazole was a reduced one, and which was further reacted with hydroxybenzoic acid to get isoxazole analog of 1,4-benzodiazepine. Again, in this reaction, we can see that dimethylamino intermediate was reacted with hydrazine to give a pyrazole analog of 1,4-benzodiazepine. And in the second reaction, the intermediate was charcoal, which was reacted with hydrazine too. But in this case, we got a reduced pyrazole analog, which was reacted with hydroxybenzoic acid to form the pyrazole analog of 1,4-benzodiazepine. In this section, we had synthesized pyrimidine incorporated analogs of 1,4-benzodiazepines linked to it directly or through a 2-phenoxyl or phenylamino spacer. Pyrimidines, they have the history of being biologically active. In view of the biological activity of pyrimidines and because of its privileged nature, we focused in our research to synthesize a new pyrimidine condensed 1,4-benzodiazepine derivatives, which will be attached on the second position through phenoxyl and phenylamino spacer. These are the compounds whose synthesis had been carried out. You can see that in this pyrimidine nuclei was incorporated through phenylamino spacer, and in all these compounds, the pyrimidine nuclei was incorporated through phenoxyl.
Hello. Hello. Ma'am, you are unmute, please. Ma'am, please unmute yourself.
Hello?
Yeah. Yes, ma'am. Yes, ma'am.
No, it just got unmute or I
No,
was.
ma'am. It's just.
Okay, okay. I thought the whole lecture got.
No, ma'am.
Okay. So should I start from here?
Yes, ma'am. Yes, ma'am.
Okay, okay. So in this section, we can see that we have the amidin intermediate, so phenylamine and peroxide spacer, which was reacted with the acetylene dicarboxylate to synthesize the pyrimidine incorporated analogs of 1,4-benzodiazepine on the second position through phenylamine and peroxide spacer. Over here we can see we had taken dimethylamino-ketone intermediate, which was reacted with urea to synthesize the pyrimidine analog again. Similarly, when dimethylamino intermediate, we see the three reactions over here. In all of these three reactions, you can see the intermediate is same, dimethylamino ketone. So second one, third one. And when they are reacted with thiourea in first reaction, guanidine in second reaction, and over here is stamidine with third reaction. All these three reactions, they will give us differently substituted pyrimidine analogs of 1,4-benzodiazepine through the phenoxyl spacer. In this reaction, we can see that we had used the chalcone intermediate. When we had reacted chalcone intermediate with urea, we got pyrimidine analog, but the pyrimidine was reduced to 1 value. That's why we had to oxidize the pyrimidine with iodoxybenzoic acid. In this, you can see that we had reacted chalcone intermediate with thiourea. In first reaction, it was with urea, now it is with thiourea. Again, a reduced pyrimidine ring was formed, and which was oxidized with iodoxybenzoic acid. So in both the reactions, we got the pyrimidine analog of 1,4-benzodiazepine through the phenoxyl spacer with different substituent on the pyrimidine ring. Over here, we had taken the chloro-substituted analogs of 1,4-benzodiazepine, and in this slide we can see that we had incorporated the different fragments of atrazine on the 1,4-benzodiazepine nuclei. In first reaction, we can see that we had incorporated aminopyrimidine on the second position. In second reaction, we had incorporated two fragments of atrazine. One is aminopyrimidine and other one is a paracyanophenoxate. In third reaction, again we have incorporated two fragments of atrazine on 1,4-benzodiazepine. The one is aminopyrimidine and the other one is paracyanophenoxate. And in the last reaction, we have incorporated all the three vital fragments of atrazine on 1,4-benzodiazepine. One hello.
Yes, ma'am. Yes, ma'am.
Yeah, actually my call got dropped. I'm sharing it again.
Okay. It's okay, ma'am.
Is it visible?
Yes, ma'am. Yes, ma'am. It's visible.
Okay, thank you. In the last section, we had synthesized benzodiazepinone, benzodiazepinone, and benzoxazepine incorporated analogs of 1,4-benzodiazepines linked to it through 2-phenyl bridge. In view of the interesting biological properties shown by azepine and 1,4-benzodiazepine, uh, it was of interest for us to design the molecules in which these moieties, that is azepine and 1,4-benzodiazepine, will be present in single molecular frame to check whether they are going to provide an additive effect on the overall biological potency in the formed molecules. And in this section, we had incorporated 1,5-benzodiazepine, 1,5-benzodiazepine, and 1,5-benzoxazepine nuclei on the 1,4-benzodiazepine, on the second position of 1,4-benzodiazepine through phenoxyl spacer. We have synthesized these different compounds over here. In red one, you can see we had incorporated 1,5-benzodiazepine nuclei, and in pink one, we can see that we have incorporated 1,5-benzoxazepine, and in the blue one, we had synthesized 1,5-benzodiazepine incorporated analogs of 1,4-benzodiazepine. All these compounds were through the phenoxyl space. To start with, you can see in these three reactions, we had started with the dimethylaminophotone intermediate, which was reacted with, in first reaction when it was reacted with orthophenyldiamine, it gave us 1,5-benzodiazepine analog, and when in second reaction it was reacted with orthoaminothiophene, it gave us 1,5-benzodiazepine analog of 1,4-benzodiazepine. And when it was reacted with orthominophenol, it gave us a 1,5-benzoxazepine phenol analog of 1,4-benzodiazepine. Similarly, when charcoal was reacted with orthophenyldiamine, it gave us 1,5-benzodiazepine analog, which was a reduced one, and which was again oxidized with isoxybenzoic acid. And when this charcoal intermediate was reacted with orthoaminothiophene, it gave us a 1,5-benzodiazepine, the reduced analog, which was oxidized over here with iodoxybenzoic acid to get the 1,5-benzodiazepine incorporated analog of 1,4-benzodiazepine through the phenoxyl spacer. And over here we had charcoal intermediate, which was reacted with orthoaminophenol to get our 1,5-benzoxazepine incorporated analog, and the reduced one over here, and then it is oxidized with iodoxybenzoic acid. These were some of the molecules that we had synthesized in our work, but the organic chemistry, in organic chemistry we see the scope is quite unlimited, and it always surprises us. The way we do organic chemistry and organic chemistry we do these days, both of these are changing. And the organic chemistry is applicable in almost all fields of our life, whether it is in fertilizers, energy, petrol, gas, medicine, polymers, textiles, or in daily life like soaps, detergent, and perfumes. Everywhere we can see there is an organic chemistry. And now, being an organic chemist, we have some future challenges. Like organic synthesis has already made a great contribution for the development of material and biological sciences, and even we can expect an increase in coming future. But on the other hand, for a sustainable future society, it is of utmost importance that we replace the traditional chemical industry towards a modern one, which is environmentally acceptable, which is a green one and clean one, and a chemistry which is in harmony with the society. So this is the responsibility of synthetic organic chemists to do better for the better life of humanity. Thank you so much.
Thank you so much, ma'am, for your enlightening lecture, your discussion on the intricate connections between chemistry, reactions, and their correlation with medicines and biologicals was incredible, insightful. despite your busy schedule and the time difference from the USA, you dedicatedly delivered a talk at midnight in the midst of winter. Your commitment to research truly shines through. Thank you so much, ma'am. Thank you so much again.
Most welcome. Thank you. Thank you.
Uh, Now I would like to invite Ms. Diksha Sharma. Please invite Dr. Seema Makhija, ma'am. Ms. Diksha Sharma.
Good afternoon, everyone. I would like to invite Dr. Siva Vakila. He is an associate professor in the Department of Zoology at the University of Delhi, Acharya Narendra Devi College. She has been an associate professor at the college for 13 years and has held that post since 2007. She previously worked as an assistant professor at same college for 4 years. Dr. Makhija also worked as an adjunct assistant professor at SGTB Khalsa College and Miranda House. Her scholastic background includes Bachelor's of Science with honors in zoology from University of Delhi in 1992, Masters of Science in Zoology from University of Delhi in 1994, and holds a PhD degree from University of Delhi in 1998. Dr. Makhija's administrative achievements are remarkable, including her work as DBT Star College coordinator and involvement on the editorial committee for the college magazine, annual report, and prospectus. She has served as a teacher in charge and has been an active member of the college editorial committee and proctoral committee. Cell biology and ciliates are her area of interest and specialization. Dr. Makhija teaches her students about evolutionary biology, cellular biology, and molecular biology. She has also supervised countless undergraduate students in college research projects through IEDC and ELITE. Her PhD supervision includes being the primary supervisor of 2 PhD students and co-supervisor for 2 others, with a focus on taxonomy, phylogeny, molecular systematics of ciliates as cellular tools to access heavy metal toxicity, microorganisms as bioindicators, and ciliates in ecotoxicological studies. Over to you, ma'am.
Thank you, Diksha, for the nice introduction. So, should I share my slides?
Sure, ma'am.
Is it visible, beta?
Yes, ma'am, it's visible.
I'm now just putting it on the slideshow. I hope it moves. Just tell me if it is not moving, huh? Is it?
Okay, ma'am.
Okay. So I must thank the organizers for inviting me to deliver this talk. I am working as professor in Department of Zoology in Acharya Narendra Devi College, and I thank all of you for inviting me to deliver this talk. The Ciliate Biology Lab was established in Acharya Narendra Dev College along with Professor Ravi Tuteja, and our group is working on several aspects like ciliate diversity, barcoding ciliates, and on the use of ciliates as biomarkers for detecting environmental pollution. Today I am going to talk about the ecotoxicological risk assessment of heavy metals in ciliates by using cellular, molecular, and bioinformatic approaches. Is it moving, Diksha?
No, ma'am.
Yes, ma'am.
Please
Yes,
try
ma'am.
again.
So if I'm coming to the second slide, it is showing on the screen. Actually, I can't see.
No, ma'am. This is first slide.
So it's not moving?
No, ma'am.
Is it visible now?
Yeah, it's ma'am. Now visible.
So should I do the same way with this type? Okay.
Yes, ma'am. Yes.
Okay. So ciliates are extremely versatile unicellular eukaryotic microorganisms. So they inhibit variety of biotypes and play a major role in regulating microbial food web. They are extremely useful model system in the studies of cell biology, molecular biology, genetics, ecology, and evolution. The journey of ciliates as model system in the field of genetics and molecular biology begin with the ciliate. All of you must be familiar with the name tetrahymena, and most of the important discoveries like the groundbreaking discoveries like catalytic RNA, telomerase and histone modifying enzymes have been conducted using ciliates as model system. So ciliates have been used as model system. Why? Because they are well adapted to different environmental conditions. They are found in cold to hot temperature, from clean to polluted environments, and they have been used in the ecotoxicological study because they have a shorter generation time. Most of them are non-pathogenic. They are easy to culture and maintain under laboratory conditions, and also easily available in all the ecosystem. Above all, they do not have cell wall, due to which they are more sensitive to environmental stress, which is important for any kind of toxicological studies. Since ciliates play a major role in designing the community structure living in the freshwater as well as terrestrial ecosystem, they act as a useful bioindicator in assessing the toxicity of environmental pollutants like heavy metals. And interestingly, they are single-celled organisms and they are performing all the function of the multicellular organisms. For working on multicellular organisms, we have to take ethical permissions, but we need not take all these ethical permissions with the ciliates. Okay. And then another important thing is that no other group of organisms have two types of nuclei. One somatic nuclei performing all the function of a normal life, whereas they also have a micronucleus, which is a germline nucleus and which is responsible for their sexual reproduction and as well as for regeneration of macronucleus. So it is a very special system on which we are working, and most of the people are not familiar how important this system is for ecotoxicological discoveries as well. The thirst of the present talk is that we are talking about how ciliates can be used in the toxicological studies, and one of the toxic component which is present in all the environments is heavy metals. There is a global increase in the concentration of heavy metals in the environment, majorly due to various anthropogenic resource sources, especially energy industry, municipal services, chemical industry, mining operations, combustion of fuels by urban and agricultural practices, and increased use of fertilizers. So all of them are contributing to pollute our natural ecosystems like freshwater, marine, as well as our terrestrial ecosystem. In a developing country like India, rapid urbanization and population growth has led to more number of industrialization, due to which various rivers of India are under the threat of heavy metal pollution. According to these studies conducted by Bureau of Indian Standards, the heavy metal concentrations in most of the rivers in India are found to be above the permissible limits. The most commonly detected heavy metals from various Indian rivers are cadmium, chromium, copper, iron, lead, nickel, and zinc. An extensive study on different sites adjoining River Yamuna showed that this region is severely contaminated with various heavy metals. It was observed that heavy metals like cadmium, copper, nickel, lead are present in the river Yamuna exceed the permissible limits. And for the present study, the samples are collected from the Okhla Bird Sanctuary, which is located at the Okhla Barrage over the river Yamuna. So our students go and collect the samples, and important thing is we collect the samples and we look into the diversity of ciliates. This picture downstairs is showing you how diverse the ecosystem is with this ciliates, and which you cannot see with the naked eye. You come to the lab, you observe under the microscope, and we identify the ciliates. The speciality of the lab is that this is one of the lab in India who can identify ciliates by using both morphological and molecular methods. And we have designed the techniques and tools for species identification in ciliates, and this review was published in 2019 by our group, where we have collected and how we can identify the ciliates. So we bring the ciliates to the lab, the samples to the lab, raise the clonal cultures, then put them in the Pringsham's medium, which is a medium suitable for the growth of the ciliates. Then we put boiled pieces of cabbage to promote the growth of bacteria which serve as a food source for the ciliates. Then we can identify the ciliates by using various methods, like we can identify them under the stereoscope microscope. We use DIC. DIC is available in our lab. So, and then we can look at the nucleus, because nucleus is also very important method of ciliate identification. So we do the Schiff's reagent staining to identify the shape and size of the nucleus, and then we do the silver line staining to mark the changes which are there on the ventral and the dorsal side, because on the ventral side of the ciliate there are group of cilia known as sterae, whereas on the dorsal side we have rows of cilia. So this is the way we identify the ciliate, and then certain ciliates have a silver line present, which is again specific to a particular species, and we use the silver line staining for that identification. Now, since nobody accepts only on the morphological basis the identification, so our lab is conducting the 18S rRNA gene sequencing to do the molecular identification of ciliates as well. So this is how we identify ciliates, by using the staining method, and by the molecular approach we extract the DNA from the clonal culture, do the amplification of 18S, then do the sequencing, and then the sequence gene is published and it is reported in NCBI. They give us the accession number, and then what we do is we generally do the phylogenetic analysis and secondary structure analysis, that is to do the 100% validation that to which group this particular ciliate belongs to. And the ciliates that we have selected for the present study are two important ciliates, that is we have isolated, and the reason why we have chosen these two ciliates, because these two ciliates were found to be the most abundant genera amongst the all which we have traced in the Okhla bird sanctuary, that is Tetmemna saprai NSB. And this NSB is reported by our group, and it has been published in 2020. This article has been published in 2020 in Saudi Journal of Biological Sciences. And Tetmemna saprai is characterized by the rigid and dorsoventrally flattened body, distinct dorsal and ventral structure arranged in a particular pattern, 2 macronuclear nodules and 2 to 4 micronuclei. And the screenshot is of that of the 18S ribosomal gene sequence which we have submitted to the gene bank. And the second ciliate that we have chosen for the present study is Euplotus adiculatus, and this is the Indian strain of Euplotus adiculatus, again published paper which we have published. This is an Indian strain, and it is characterized by rigid pellicle, rectangular shaped and dorsoventrally flattened body. It has a dorsal erigosome of double eurystomus type, macronucleus C-shaped with an arc at the back and having a single micronucleus. It is very clear from the diagram the structure and shape of the cell. Now to the main concern. Among the heavy metals, we have chosen cadmium and copper for the present study, because they have been detected to be present predominantly in the aquatic system along with zinc, lead, iron and manganese. Also in various ecotoxicological studies using ciliates, copper and cadmium were observed to be more toxic to ciliates as compared to other heavy metals. Thus, the two metals, that is cadmium and copper, are known to be more toxic to ciliate, have been selected for this study. Now, heavy metals induce oxidative stress. All of you know that, and I think we have 2 or 3 lectures on today which have talked about how ciliates induce oxidative stress in a group of animals. So same way, the oxidative stress is induced in the ciliates by generating increased level of ROS, that is reactive oxygen species. So same way, metals with redox activity like copper generate ROS directly by autooxidation, and metals without redox activity like cadmium generate ROS indirectly by affecting the antioxidant defense system. Okay, so to combat heavy metal toxicity, ciliates have developed many defense mechanisms like increased production of several antioxidant enzymes and stress responsive genes, mainly metallothionein and heat shock proteins. Antioxidant enzymes include superoxide dismutase, catalase, glutathione peroxidase. They are involved in detoxification of ROS and in protecting the cells from oxidative stress. Metallothioneins are also known to be involved in heavy metal bioaccumulation in ciliates. As all of you know, heat shock proteins play a major role in folding, assembly, and distribution. So due to their vital role, these HSPs are also upregulated in ciliates to combat the stress. And the cellular and molecular basis of heavy metal induced stress is again published by our group. So the objective of the present study was, which I am presenting today, that is to analyze the toxicity assay for determining the sensitivity and survival of supply and adiculators after exposure to heavy metals, that is cadmium and chromium. Then enzymatic assays were performed for studying the heavy metal induced oxidative stress, like the generation of ROS was assessed by the activities of enzymes like SOD, catalase and glutathione peroxidase. Then we also studied the expression of stress responsive genes, that is MNSOD, catalase, cytosolic SHP70 of both sapri as well as adiculators. We characterize for the first time, it is not that we characterize for the first time, this characterization was done for the first time in these two ciliates, and it was done by our ciliate biology group in Delhi, India. So heavy metals, which are commonly released into the aquatic ecosystem, as we have told you earlier, they are majorly from cigarette smoking, e-waste, fertilizers, fuel combustion, incineration, mining companies, etc. So number of earlier studies have shown that heavy metals have adverse effect on growth rate of ciliates. In various reports, it was shown that percent survivability of ciliates decreased significantly with increasing heavy metal concentration. So therefore, toxicity assay performed by us to determine the lethal and sublethal effect of cadmium and copper on Tetmemna sapri and Euplotus adiculatus. For this, cells of sapri and adiculatus were treated with cadmium and copper ranging from 1 to 5 mg per liter and 0.5 to 0.1 to 0.5 mg per liter respectively. The LC50 for cadmium was 2.44 mg per sapri and 2.24 mg per liter for adiculatus. And the LC50 for copper was found to be 0.14 mg per liter in sapri and 0.18 mg per liter in adiculatus. So in present study, both the ciliates were found to be more sensitive to copper, showing that copper is more toxic as compared to cadmium in the case of ciliates. Okay, so next thing that we did was, that is cadmium and copper, as we have already told you, they are known to induce, but nobody has given the proof. So we are the ones who have given the proof that activity of ROS scavenging enzymes were evaluated role in the stress response towards heavy metals like cadmium and copper. So the first enzyme activity which we have studied is superoxide dismutate. SOD is the first line of defense against ROS, which catalyzes the superoxide radicals to produce hydrogen peroxide. Concentration of SOD in sapri as well as adiculatus increase with increase in heavy metal concentration. But as you can see in the graph, at LC70 level, the concentration of SOD decreased moderately, though significantly higher than the control. It suggests active participation of SOD in detoxifying superoxide radicals which are generated by exposure to heavy metal. Since SOD is known to protect the cells from oxidative damage, it shows higher activities shows that cells tolerance towards heavy metal stress. In both the ciliates, a slight drop in SOD at LC70 dose but was significantly higher. This could be because of the inhibitory effect of these heavy metals on the enzyme activity at higher concentrations. The second enzyme that we have studied is catalase. It also showed the same thing, that the activity increased but slight decrease we have observed as compared to that of, but the level is more as compared to that of control. It is very clear from the graph. So cadmium is known to induce toxic effects by binding to the sulfhydryl group of the heme present in the active site of the catalase enzyme, thereby lowering the enzyme activity. Under normal condition, SH group present in the cat enzyme gets oxidized to SS group in the presence of hydrogen peroxide, which is reduced to SH group with the help of cofactor NADPH. But cadmium binding inhibits the conversion of SS group to SH, thereby inhibiting the catalytic function of this enzyme. Also, copper is known to inhibit the same way, but cadmium being non-essential and redox inactive metal, the inhibition rate is seen more predominantly as compared to copper. So the next enzyme that we have studied is glutathione peroxidase. It increased in both the chelates with the increase in the heavy metal concentration. And interestingly, in Euplotus adiculatus, GPX concentration increased steadily with the increase in the metal concentration. So cadmium normally affects the enzyme activity by binding to the SH group of glutathione or by directly binding to the active site of the GPX enzyme. And these results indicate that GPX have selenocysteine group in the active sites, making the enzyme more promising enzyme towards the detoxification of the ROS, which is generated by the heavy metal stress. So our work never ended here. We wanted to know the response of the expression of the stress responsive genes. So far we have worked with the enzymes. Now we wanted to know what is happening at the level up. That is, the regulation is happening at the level where there is increase in the transcription of these enzymes, and we went into that. We isolated the total RNA both from Tetmemna sapri as